Hi everybody. So in the competitive classes today let me start the chapter biotechnology principles and processes. Okay. So let the question number 1. So plasmid has been used as a vector because plasmid is yake vector agi use ಮಾಡ್ತೀವಿ. So both its ends show a replication. No. So both its ends, there is no term called both for plasmids since a plasmid is a circular DNA. Okay, so it is circular DNA, but eukaryotic DNA is linear. So eukaryotic DNA have the cut ends. They can be said to have both ends, but you cannot use the same term for plasmid since it don't have the both ends. Okay, so it is not the right option. It can move between prokaryotic and eukaryotic cells. No, never. So this prokaryotic plasmid which cannot move into the eukaryotic cell unless until we are forcibly introduce it. Of course, in eukaryotic cells it cannot express, so it is not the right option. It is a circular DNA, no doubt, which have capacity to join to eukaryotic DNA. Okay, so we can cut this plasmid to the cut ends. You can join the desired DNA. So desired DNA from any eukaryotic organism you can join to make it rDNA. So the first rDNA, which consisting of native plasmid of Salmonella to this antibiotic resistant gene is introduced. Okay, to make it the first rDNA. Okay, so Herbert and Boyer first did it. Then this rDNA was introduced into E. coli bacteria. So the first rDNA which was introduced, which was created. Okay, so of course the circular DNA which can capacity to join to eukaryotic DNA. So it can accept this alien DNA or foreign DNA as its own. Okay, so ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಏನು ಅನ್ಕೊಳ್ಳುತ್ತೆ, ಈ ಜಾಯಿನ್ ಮಾಡೋ ಡಿ ಎನ್ ಎನು ಕೂಡ ನಂದೇ ಅಂದ್ಕೊಂಡು ಇಟ್ ಸ್ಟಾರ್ಟ್ಸ್ ರೆಪ್ಲಿಕೇಟೆಡ್. Okay, so it is one of the important property of this plasmid that it can accept the foreign DNA and replicate it. Okay, so it has one ovocyte. With the help of that ovocyte it starts replicate it. Okay, and it also start to produce the product. ಓಕೆ, ಇದು ಕೂಡ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಿ ಇದು ಕೂಡ ಪ್ರಾಡಕ್ಟ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋದಷ್ಟು ಅಕ್ಸೆಪ್ಟ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ. ಇಟ್ ಕೆನಾಟ್ ರಿಜೆಕ್ಟ್ ಎನಿ ಫಾರಿನ್ ಡಿ ಎನ್ ಎ. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಇಂಪಾರ್ಟೆಂಟ್ ಪ್ರಾಪರ್ಟಿ. ಸೊ ದಟ್ ಇಸ್ ದ ರೀಸನ್ ವೈ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಯೂಸ್ ಇಟ್ in rDNA technology. So it has antibiotic resistant gene. Remember, not all the plasmids, not all the native plasmids have antibiotic resistant gene. Actually these antibiotic resistant genes are introduced into the plasmids. ಪ್ಲಾಸ್ಮಿಡ್ಸಿಗೆ ಬೇಕಂತಾನೆ ಸೈಂಟಿಸ್ಟ್ಗಳು ಅದನ್ನು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರ್ತಾರೆ. ಓಕೆ, ಸೊ ವೈ ದೇ ಆರ್ ಇನ್ಸರ್ಟೆಡ್ ದಿಸ್ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಅಂದರೆ ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಯೂಸ್ಡ್ ಆಸ್ ಸೆಲೆಕ್ಟಬಲ್ ಮಾರ್ಕರ್ ಆಗಿ ಯೂಸ್ ಮಾಡಲಿಕ್ಕೆ ಅಂತಾನೆ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿಸಿರ್ತಾರೆ. ಸೊ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇನ್ ಕೇಸ್ ಆಫ್ PBR322, two antibiotic resistant genes were there. One is ampicillin resistant gene, another one is a tetracycline resistant gene. Okay, so these resistant genes were initially not present. Okay, so they were inserted from other organisms. Okay, so they got the ability to resist that particular antibiotics, and by using this ampicillin tetracycline resistant gene we can easily select the transformins from non-transformins. Okay, that is the reason why this rDNA, for example PBR322 is considered as a tailored plasmid. ಇದನ್ನು ನಾವು ಟೈಲರ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಟೈಲರ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್ ಅಂದರೆ ನಮಗೆ ಏನೇನು ಬೇಕಿದೆಯೋ ಕ್ಯಾರೆಕ್ಟರ್ಸ್, ಆ ಕ್ಯಾರೆಕ್ಟರ್ಗಳನ್ನೆಲ್ಲ ಜಾಯಿನ್ ಮಾಡ್ಕೊಂಡಿರುವಂಥದ್ದು. ಓಕೆ, ಸೊ ದಟ್ಸ್ ವೈ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಟೈಲರ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್. So based on this, again, no doubt, of course it has the antibiotic resistant gene. Okay, it has the antibiotic resistant gene, but this is not the right answer since because of that antibiotic resistant gene only we are not going to use as a vector. Antibiotic resistant gene ಇದೆ ಅಂತ ಮಾತ್ರಕ್ಕೆ ನಾವು ಅದನ್ನು ವೆಕ್ಟರಾಗಿ ಯೂಸ್ ಮಾಡಬೇಕಂತ ಇಲ್ಲ. ಓಕೆ, ಸೊ ವಿತೌಟ್ ದಿಸ್ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಆಲ್ಸೋ we can use it as a vector since it can join the desired DNA from other source, other organism. Okay, some plasmids instead of antibiotic resistant genes as a selectable marker, some have like a PUC18. plasmid of University of California, PUC18, has, instead of antibiotic resistant gene, it has the gene called a LAC-Z gene. It has a LAC-Z gene. It can produce a beta galactosidase. That beta galactosidase can act as a chromogenic selectable marker. When beta galactosidase is produced by this gene, it can produce the blue color colony. Here if you cut and join any other alien DNA or foreign DNA, so it lost the ability to synthesize beta galactosidase. So transformants give white color colony, non-transformants give the blue color colony. So instead of this antibiotic resistant gene also, plasmid can exist and can be used as a selectable marker by different genes, okay, like LAC-Z gene in case of PUC18. Okay, so C is the right option for this question. Okay. Move to the next question. So, which of the following is known as a specific molecular scissor? Okay, so yaudana navu molecular scissors anta karitive. Okay, so molecular scissors. Okay, why they are called so? Let's see. So ligase is an enzyme, helicase is an enzyme, restriction endonuclease is an enzyme, DNA polymerase is an enzyme. So all are these are enzymes. All the options contains the enzymes only. Then let's see the functions one by one. So ligase is an enzyme. It is called as molecular glue. Idana molecular glue anta karitive. Andre it can join the molecules. It can join the molecules. Okay. So it create a phosphodiester bond. Okay, ligase ಏನು ಮಾಡುತ್ತೆ phosphodiester bond ಅನ್ನು create ಮಾಡುತ್ತೆ. So hope you remembered that Okazaki fragments which are produced during DNA replication. So here RNA primers were removed and joining of these cuttings of newly synthesized DNA is carried out by this ligase enzyme. So ligase enzyme can create phosphodiester bond. Okay, so ligase is an enzyme that can create the phosphodiester bond, so it is called as molecular glue. It can be used as stitcher. Of course, it is also called as a molecular stitcher. It is a molecular stitcher. It can join the cuttings. Okay, so that is a function of ligase. Then helicase. So helicase is an enzyme that can unwind the DNA. So during DNA replication, so this DNA should be unwinded. Okay, so DNA unwind agi replication start agbeku. So that unwinding enzyme ee riti ನೋಡಿ DNA unwind ಆಗಬೇಕು, ಈ ರೀತಿ ಓಪನ್ ಆಗಬೇಕು, ಒಂದು complete pitch open ಆಗಬೇಕಂದ್ರೆ DNA should be unwinding. So that unwinding enzyme is called as helicase. Unwinding enzyme is helicase. It is also called as topoisomerase 1. Topoisomerase 1. Okay, similarly topoisomerase 2 is also there that remove the super twists or super coilings which are caused during DNA unwinding. Okay, so helicase, it is unwinding enzyme. Then restriction endonuclease, they restrict something they have inside the nucleus, so it related to nuclease or with related to DNA, so called as a restriction endonuclease. So why the name endonuclease, restriction endonuclease? Okay, so they restrict what? So you are familiar with the hyperparasite. Okay, you are familiar with the hyperparasite. So hyperparasite is a parasite which parasitize the parasite, called hyperparasite, like bacteriophage. ಈ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ಗಳನ್ನು ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳಿಗೆ ಇನ್ಫೆಕ್ಟ್ ಮಾಡುವಂಥ entities are called as bacteriophages. Okay, so these are the viruses. Bacterial infecting phage. Phage means virus. Okay, so bacteriophage. So when the bacteriophage infect any bacteria by inserting its central core, by contracting the sheath, okay, it inject the genetic material. Okay, so genetic material of bacteria. So this DNA, inserted viral DNA, can join to, can become an integral part of this bacterial DNA, and it starts synthesize viral protein and viral genetic material, that is viral DNA synthesize. So ಏನು ಮಾಡುತ್ತೆ, ಇದು ಇನ್ಸರ್ಟ್ ಮಾಡಿದ ನಂತರ ಅದು ಹೋಗ್ಬಿಟ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಜೀನೋಮ್ ಒಳಗೆ ಸೇರ್ಕೊಳ್ಳುತ್ತೆ. ಸೊ ಇಟ್ ಆರ್ಡರ್ ದಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್, ನೋಡಿ ಒಳಗೆ ಬಂದು ಸೇರ್ಕೊಂಡ್ಬಿಟ್ಟು ಇಟ್ ಆರ್ಡರ್ ಮಾಡುತ್ತೆ. ಸೊ ಸ್ಟಾರ್ಟ್ ಸಿಂಥಸೈಸ್ ವೈರಲ್ ಪ್ರೋಟೀನ್ ಆ್ಯಂಡ್ ವೈರಲ್ DNA ಅಂತೇಳಿ. ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸ್ಟಾರ್ಟ್ ಸಿಂಥಸೈಸ್ ವೈರಲ್ DNA ಆ್ಯಂಡ್ ವೈರಲ್ ಪ್ರೋಟೀನ್. ಫೈನಲಿ ನ್ಯೂ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜಸ್ ವರ್ ಫಾರ್ಮ್ ಆ್ಯಂಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ is get rupture and they are released into the outside. Okay, so to avoid this, some bacteria have enzymes. Those enzymes are called as endonuclease enzymes or restriction enzymes. So adhen madutte, these enzyme cut the viral DNA into fragments. ಈ ರೀತಿ ಚಿಕ್ಕ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ಗಳನ್ನಾಗಿ ಕಟ್ ಮಾಡಿಬಿಡುತ್ತೆ. ಸೊ ಈವನ್ ದೋ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಇನ್ಸರ್ಟ್ಸ್ ದ viral DNA, it is unable to bind to bacterial genome because it is made into small fragments. So those enzymes which have the ability to cut the DNA into fragments are called as endonuclease enzymes. So since they restrict the effect of bacteriophage DNA, so they are called as restriction endonuclease. Okay, so this is a group of enzyme, so we have the ability to cut the DNA. So ಇಲ್ಲಿ restriction enzymes has two types. Okay, so restriction enzymes has two types. One is restriction endonuclease, and another one restriction exonuclease. So what is the difference between these two? Remember, both are restriction enzymes only. So let's see the difference. So this is the DNA, this is also double stranded DNA. Okay, so here restriction endonuclease can make a cut at any point on a DNA. ಅಂದರೆ ಯಾವುದೇ ಜಾಗದಲ್ಲಿ ಅಫ್ಕೋರ್ಸ್ ದೇ ನೀಡ್ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್. ನೋಡಿ ಇಲ್ಲೊಂದು ಕಟ್ಟು ಈ ರೀತಿ ಮಧ್ಯದಲ್ಲಿ ದೇ ಕ್ಯಾನ್ ಕಟ್ ದ DNA ಇನ್ ಬಿಟ್ವೀನ್ ವೆರಾಸ್ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಕ್ಯಾನ್ ಕಟ್ ಅಟ್ ದಟ್ ಎಂಡ್ಸ್ ಓನ್ಲಿ. DNA ನ ಎಂಡ್ ಎಲ್ಲಿರುತ್ತೆ ಆ ಎಂಡಲ್ಲಿ ಮಾತ್ರ ಕಟ್ ಮಾಡೋ ಎಬಿಲಿಟಿ ಇರುತ್ತೆ. Okay, they are unable to cut the DNA in between. Okay, so that is the reason why there are two types of enzymes, restriction endonuclease and restriction exonuclease. So usually this restriction endonuclease, we call it as a restriction endonuclease. So restriction enzymes in a short form. So RNA ಜಾಸ್ತಿ ಯೂಸ್ ಮಾಡ್ತೀವಿ biotechnology because we need to cut the DNA at in between. Okay, most of the time we need to cut the DNA in between, so we are going to use restriction endonuclease enzymes. Okay, so this restriction endonuclease enzyme cut the DNA at a specific point. So okay, ಎಲ್ಲೆಲ್ಲಿ ಬೇಕು ಅಲ್ಲೇ ಕಟ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ, they need a specific sequence. That specific sequence is called as restriction sequence ಅಥವಾ restriction site, called as restriction sites or cloning sites. ಈ ಥರ ಬೇರೆ ಹೆಸರುಗಳಿದೆ. ಓಕೆ, ಸೊ ಎಲ್ಲದೂ ಕೂಡ ಒಂದೇ. ಆ್ಯಂಡ್ ದೆ ನೀಡ್ ಎ ಸ್ಪೆಸಿಫಿಕ್ palindromic ಸೀಕ್ವೆನ್ಸ್ ದೆ ನೀಡ್. ಇದನ್ನು ಕಟ್ ಮಾಡಬೇಕಂದ್ರೆ ಅವಕ್ಕೆ palindromic ಸೀಕ್ವೆನ್ಸ್ ಬೇಕಾಗತ್ತೆ. ಸೊ palindromic ಸೀಕ್ವೆನ್ಸ್ ಅಂದರೆ ಆ್ಯಂಡ್ ಬೋತ್ ದ ಡೈರೆಕ್ಷನ್ ದೆ ಸೇಮ್, ಒಂದೇ ಥರ ಓದುವಂಥ ಒಂದೇ ಥರ ಇರುವಂಥ ಇದು. ಓಕೆ, ಯು ಆರ್ ಫೆಮಿಲಿಯರ್ ವಿತ್ ದ ಒನ್. ಇಟ್ ಈಸ್ GAATTC. GAATTC. ಈ ಕಡೆಯಿಂದನೂ GATTC, ಈ ಕಡೆಯಿಂದನೂ GATTC. ಓಕೆ, ಸೊ ದೋಸ್ ಕೈಂಡ್ ಆಫ್ sequences are called as palindromic sequence. Each and every restriction enzyme need a particular palindromic sequence. On DNA, wherever that palindromic sequence is there, they go and cut the DNA. Okay, so since they are able to cut the DNA, the function of scissor is to cut. Okay, so they can cut the DNA. Since they are at the molecular level, it is called as molecular scissors. Okay, so the right option is this one. Then a DNA polymerase enzyme. You know, DNA polymerase enzyme are the enzymes that can polymerize the synthesis of DNA. They can join the nucleotides one by one by creating the phosphodiester bond to make DNA a long chain of polynucleotide chain. So that is the function of DNA polymerase. So DNA polymerase has 3 types: DNA polymerase 1, DNA polymerase 2, similarly DNA polymerase 3. So 3 types of polymerase enzymes were there. Among these, DNA polymerase 3 is the main enzyme. DNA polymerase 3 is the main enzyme that can polymerize the DNA, whereas this DNA polymerase 1 and DNA polymerase 2, they act as a proofreaders. Okay, they act as a proofreaders, and in the absence of DNA polymerase 3, DNA polymerase 2 can act as a polymerase enzyme. Okay, so proofreading means, so during DNA replication, if any nucleotide added wrongly, that can be removed, and actual DNA nucleotide was added during that proofreading. So those proofreading enzymes are DNA polymerase 1 and DNA polymerase 2. Okay. So this is about the enzymes, different kinds of enzymes. So similarly, you studied one thermostable polymerase enzyme, DNA polymerase enzyme. Okay, that is called a Taq polymerase, which was used in PCR. Okay, yeah. Move to the next question. The enzyme used for joining 2 DNA fragments. Okay, so which have sticky ends. Suppose this is one DNA which having unpaired bases. Okay, so unpaired bases iruantaddu sticky end anta heltive. So let's say this is one sticky end. Write it different color. 1, 2, 3, 4. 1, 2, 3, 4. So this is the fragment of DNA. Just imagine this is a circular DNA like a plasmid. So ee reeti ide circular DNA. So these are also have the unpaired base. Here also unpaired bases were there. Here also unpaired bases were there. Here also unpaired bases were there. Okay, if such unpaired bases are available, so these can, since remember it should be have the complementary bases for this. Complementary base pairing ago tara idre, if they have complementary bases, they can be joined together. So hege andre, so it can be joined together. 1, 2, 3, 4. Then can be continued. Okay. Now it can be 1, 2, 3, 4. Can continue like this. Okay, so ಈ ಥರ ಎರಡು DNA ಜಾಯಿನ್ ಮಾಡ್ಬೋದು. ಈ ರೀತಿ ಜಾಯಿನ್ ಮಾಡಬೇಕು ಅಂದರೆ ಸೀದೆ ಇಲ್ಲೇನಾಗಿರಬೇಕು, ಎರಡು ನೋಡಿ ಕಟ್ ಎಂಡ್ಸ್ ಇರುತ್ತೆ, ಇದನ್ನು ಸೀಲ್ ಮಾಡಬೇಕು. ದೇ ಶುಡ್ ಬಿ ಸೀಲ್ ಹಿಯರ್, ದೆನ್ ಹಿಯರ್, ಹಿಯರ್, ಹಿಯರ್. ದ ಸೀಲಿಂಗ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ creating phosphodiester bond. So creation of that phosphodiester bond is done by the enzyme called a DNA ligase. Okay, so remember that a restriction endonuclease break this phosphodiester bond. So it breaks the phosphodiester bond and remove that part of the DNA, whereas the ligase enzyme create the phosphodiester bond between the cut ends of DNA. Okay. So these are called molecular glues. Restriction endonuclease we are already discussed. Then DNA polymerase we discussed. Then gyrase. So gyrase is nothing but topoisomerase 2. So gyrase is an enzyme which removes super twists. It removes super twists or super coiling. Super coiling. So this super coiling is formed when this DNA is get unwinded. So hope you understand this. Okay. So when DNA is get unwinded due to the function of helicase enzyme, suppose ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿಂದ DNA ಅನ್ವೈಂಡ್ ಆಗಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಯಿತು ಅಂತ ಅಂದ್ಕೊಂಡ್ರೆ ನೋಡಿ, DNA ಈ ಥರ ಇದಾಗ್ತಾ ಇರುತ್ತೆ, ಅದು ಅನ್ವೈಂಡ್ ಆಗಬೇಕಾದ್ರೆ ಈಗ ಆಗ್ತಾ ಇರುತ್ತೆ. ಸೊ ಈ ರೀತಿ ಆಗ್ತಾ ಇರಬೇಕಾದ್ರೆ ಇಟ್ ಕ್ರಿಯೇಟ್ ದ ಟೆನ್ಷನ್ ಆ್ಯಂಡ್ ಫ್ರಮ್ ಹಿಯರ್ ಆನ್ವರ್ಡ್ಸ್ ಇಟ್ ಸ್ಟಾರ್ಟ್ಸ್ ಕಾಯಿಲಿಂಗ್ ಎಗೇನ್. ಸೊ ಈ ರೀತಿ ಗಂಟು ಆಗಿಬಿಡುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ಅದು ಟೆನ್ಷನ್ ಕ್ರಿಯೇಟ್ ಆಗಿದ್ದು ಇಟ್ ಮೇಕ್ ದ DNA ಇಂಟು ಈ ರೀತಿ ಜಾಯ್ನ್ ಆಗುತ್ತೆ. ಸೊ ಯು ಕ್ಯಾನ್ ಡೂ ದಿಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಬೈ ವಿತ್ ಅ ಥ್ರೆಡ್ನ ತೊಗೊಂಡು ಒಂದು ಕಡೆ ಹಿಂಗೆ ಓಪನ್ ಅಪ್ ಮಾಡ್ತಾ ಹೋದೆ ಅಂದರೆ 2 ಲೈನ್ ಇದಿರೋವಂಥದ್ದು DNA ಥರನೇ ಇರುವಂಥ ಅದರ ಒಂದು ವೈರ್ನ ಅಥವಾ ದಾರನ ತೊಗೊಂಡು ಹಿಂಗೆ ಹೇಳಿದ್ರೆ ಒಂದು ಕಡೆ ಎಳಿತಾರೆ, ಇನ್ನೊಂದು ಕಡೆ ಗಂಟು ಸುತ್ಕೊಂಡ್ಬಿಡುತ್ತೆ. ಸೊ ದೋಸ್ ಆರ್ ಕಾಲ್ಡ್ ಸೂಪರ್ ಕಾಯಿಲಿಂಗ್ಸ್. ಸೊ ಅದನ್ನು ರಿಮೂವ್ ಮಾಡುವಂಥದ್ದು ಗೈರೇಸ್ ಎನ್ಸೈಮ್. ಓಕೆ. ಸೊ ವೈಲ್ ಅನ್ವೈಂಡಿಂಗ್ ದ DNA ಹೆಲಿಕೇಸ್ ಎನ್ಸೈಮ್ ಕಟ್ ಓನ್ಲಿ ಒನ್ ಎಂಡ್, ಅಂದರೆ ಒಂದು ಫಾಸ್ಫೋಡೈಸ್ಟರ್ ಬಾಂಡ್ನ ಕಟ್ ಮಾಡಿ ಈ ಕಡೆಗೆ ತೊಗೊಂಡು ನೋಡಿ, ಅದನ್ನು ಈ ಕಡೆಗೆ ತೊಗೊಂಡು ಇಟ್ ಕ್ಯಾನ್ ರೈಟ್ ಕ್ಲಿಯರ್ಲಿ. ಓಕೆ. ಸೊ ಅದೇನು ಮಾಡುತ್ತೆ, ಇಲ್ಲಿ ಸಪೋಸ್ ಇಟ್ ಮೇಕ್ ಎ ಕಟ್ ಹಿಯರ್ ಅಂತ ಅಂದ್ಕೊಂಡ್ರೆ ಇದನ್ನು ಈ ಕಡೆಗೆ ತೊಗೊಂಡು ಇಟ್ ಕ್ರಿಯೇಟ್ ದ ಡೈಎಸ್ಟರ್ ಬಾಂಡ್. ಅದನ್ನು ಈಗ ತೊಗೊಂಡು ಇಲ್ಲಿ ಮತ್ತೆ ಜಾಯಿನ್ ಮಾಡುತ್ತೆ. ಸೊ ಈ ರೀತಿ ಇರೋದನ್ನು ತೊಗೊಂಡು ಒಂದು ಕಡೆ ಇದನ್ನು ಕಟ್ ಮಾಡಿ ಮತ್ತೆ ವಾಪಸ್ ಹಿಂಗೆ ತೊಗೊಂಡು ಅದನ್ನು ಜಾಯಿನ್ ಮಾಡುತ್ತೆ. ಸೊ ಈ ವೈರಾಸ್ ಗೈರೇಸ್ ಎನ್ಸೈಮ್ ಟು ರಿಮೂವ್ ದ ಸೂಪರ್ ಟ್ವಿಸ್ಟ್ ಇಟ್ ಕಟ್ ದ ಹೋಲ್ ಸ್ಟ್ರಾಂಡ್ ಆಫ್ DNA ಜಸ್ಟ್ ಲೈಕ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ ಥರನೇ ಎರಡೂ ಸ್ಟ್ರಾಂಡನ್ನು ಕಟ್ ಮಾಡಿ ವಾಪಸ್ ತೊಗೊಂಡು ಹಿಂದೆ ಮತ್ತೆ ಜಾಯಿನ್ ಮಾಡುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ಗೈರೇಸ್ ಎನ್ಸೈಮ್ ಕಾಲ್ಡ್ ಆಸ್ topoisomerase 2. ಹೆಲಿಕೇಸ್ ಎನ್ಸೈಮ್ is topoisomerase 1 whereas gyrase is topoisomerase 2. Okay. So the enzyme used to join the two ends of DNA is ligase. Okay. Next move on to the next question. Question number 4. Okay. So restriction endonuclease HIND2 always cut the DNA molecule at a particular point by recognizing a specific sequence. ಸೊ ಆ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸಲ್ಲಿ ಎಷ್ಟು ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ? 6 ಬೇಸ್ ಪೇರ್ಸ್, 5 ಬೇಸ್ ಪೇರ್ಸ್, 4 ಬೇಸ್ ಪೇರ್ಸ್, 7 ಬೇಸ್ ಪೇರ್ಸ್. ಸೊ ಇನ್ ಮೋಸ್ಟ್ ಆಫ್ ದ ಟೈಮ್, ಸೊ ದಿಸ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಎನ್ಸೈಮ್ ಹ್ಯಾಸ್ 6 ಬೇಸ್ ಪೇರ್ಸ್. ಓಕೆ. ಸಪೋಸ್ ಫೆಮಿಲಿಯರ್ ಇರುವಂಥ GAATTC ಫಾರ್ EcoR1. So it also has 6 base pairs. So similarly HIND2. So HIND2 is the first restriction enzyme. It was isolated and named, and it has the recognition sequence. So it has a particular recognition sequence to cut it. So it has the recognition sequence AAGCTT. Sorry, AAGCTT. So it's the recognition sequence for HIND2. Okay. So AAGCTT. Okay. So ಈ ರೀತಿ recognition sequence ಇರುತ್ತೆ. So it can make a cut between A and A in both the direction. Okay. Whereas in case of EcoR1 it make a cut between G and A, whereas HIND2 cut between A and A. Okay. So by breaking the phosphodiester bond first, then it can remove hydrogen bond. Okay. So this is how restriction enzyme nuclease enzyme cut the DNA. So here phosphodiester bond is broken by this enzyme, whereas the ligase enzyme create this bond again. Okay, during joining of DNA. Okay. So this is about the restriction enzyme into 2. So Haemophilus influenzae strain D is the full form of IND2. Okay. So Haemophilus influenzae. Haemophilus influenzae strain D. Okay. So Haemophilus influenzae, Okay, so you know the naming of a restriction enzyme. So here that first letter stands for genus name, species name, strain name. Okay, and this is the order of the enzyme isolated from that particular organism. Okay, so why the name IND2 for the first restriction enzyme? Of course they isolated two enzymes, IND1 and IND2, but for IND2 enzyme they confirmed that it has the property of cutting the DNA at a specific sequence, so it acts as an endonuclease. So later they found that IND1 also has the same property. Okay. Yeah. Next move on to the question number 5. So DNA fragment generated by the restriction endonucleases in a chemical reaction can be separated by polymerase chain reaction, electrophoresis, restriction mapping, centrifugation. So ಯಾವ ಮೆಥಡ್ ಇಂದ ನಾವು DNA by restriction enzyme ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಕಟ್ ಮಾಡಿರ್ತೀವಿ, ಆ ಕಟ್ ಮಾಡಿರುವಂಥ ಸ್ಟ್ರ್ಯಾಂಡ್ಸ್ ಅನ್ನು ಸಪರೇಟ್ ಮಾಡಬಹುದು. ಓಕೆ, so suppose this is the DNA, it can make into several pieces for example, so like that. So it undergo fragmentation here, here and here. Okay, so we need to separate this fragment. See the length of the fragments. So ಈ ರೀತಿ ಡಿಫ್ರೆಂಟ್ ಲೆಂತ್ ಇರುವಂಥ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ಗಳು ಫಾರ್ಮ್ ಆಗಿರುತ್ತೆ. ಸೊ ರಿಮೆಂಬರ್ ದ ಲೆಂತ್ ಆಫ್ ದ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ವಿಚ್ ಆರ್ ಫಾರ್ಮ್ಡ್ ಆಫ್ಟರ್ ದ ಡೈಜೆಷನ್ ವಿತ್ ದ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್, ಇಟ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ಹೌ ಮೆನಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ಸ್ ಆರ್ ರೆಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸಸ್ ಇದೆ. ರೆಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಎಷ್ಟಿರುತ್ತೋ ಅಷ್ಟು ಫ್ರಾಗ್ಮೆಂಟ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಓಕೆ, ಇದು ನೋಡಿ, ಒಂದು ತುಂಬ ಚಿಕ್ಕದಿರೋದಿದೆ, ಅದಾದಮೇಲೆ ಇಲ್ಲೊಂದಿದೆ, ಇದೊಂದಿದೆ. ಸೊ ಲೆಟ್ ಮಿ ಗಿವ್ ದ ನಂಬರ್ ಫಾರ್ ದೆಮ್. ಫಸ್ಟ್, ಸೆಕೆಂಡ್, ಥರ್ಡ್ ವಿತ್ ರೆಸ್ಪೆಕ್ಟ್ ಟು ದೇರ್ ಲೆಂತ್, then fourth according to their length. So in case of after digestion, when you try to separate them, so we are mentioned in electrophoresis as kilo base pairs. Now ಒಂದೊಂದೇ ಫ್ರಾಗ್ಮೆಂಟ್ ತೊಗೊಂಡ್ರೆ ಹೇಳೋಕ್ಕಾಗಲ್ಲ, ಸೊ ಮಲ್ಟಿಪಲ್ ಫ್ರಾಗ್ಮೆಂಟ್ ತೊಗೊಂಡ್ರೆ ಬೇಸ್ಡ್ ಆನ್ ದ ಕಿಲೋ ಬೇಸ್ ಪೇರ್ಸ್, ಹೌ ಮೆನಿ ಬೇಸ್ ಪೇರ್ಸ್ ವರ್ ದೇರ್, ಸೊ ಬೇಸ್ಡ್ ಆನ್ ದಟ್ DNA is going to be separated. So among them electrophoresis is the one, it is extensively used in DNA fragment separation. After cutting the DNA into fragments, so those fragments, of course the whole process takes place in a small test tube. So isolated DNA is put into one test tube. To that you are going to add a restriction enzyme. So this restriction enzyme made the DNA into cuts, very fragments according to the number of recognition sequence it has. It made a cut. Now we need to separate them on electrophoresis. So in electrophoresis there were a space called wells. So while preparing this electrophoresis slab itself, we are able to prepare that holes. Okay, into that holes we are going to add the DNA. After digestion they can be add into wells. Okay, so wellige adanna add ಮಾಡಿ. So this slab, this gel electrophoresis slab is made out of agarose. Agarose is nothing but that highly purified agar, we can say. Okay, so isolated from gelidium and gracilaria. So highly purified form, that is agarose. So this agarose is act as a medium to separate these DNA fragments. Okay, so under the influence of current. Okay, so when you apply the current here, so it can be made into connect to the electrodes. Okay, so one end. So remember, so the process of separation of this DNA fragments, it also depends on property of DNA with respect to its charge. Property of DNA with respect to its charge. So you know DNA has a negative charge because of that phosphoric acid it has. Okay, so only phosphoric acid pair ಆಗಿರುತ್ತೆ ಇಲ್ಲಿಗೆ ಫಿಫ್ತ್ ಕಾರ್ಬನ್ಗೆ. in a pentose sugar it paired like this. So ಇಲ್ಲಿ ನೋಡಿ ಈ ರೀತಿ ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಇರುತ್ತೆ. So this negative charge of DNA, it is due to this phosphoric acid present there at the fifth carbon of pentose sugar. Okay, so it carries negative charge. ಸೊ ಹೀಗೆ ನೆಗೆಟಿವ್ DNA ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಇದೆ ಅಂದರೆ ಯು ಶುಡ್ ಬಿ ಲೋಡೆಡ್. ನೋಡ್ಕೊಳ್ಳಿ, ಅದನ್ನು ನಾವು ನೆಗೆಟಿವ್ ಎಂಡ್ ಕಡೆನೇ ಲೋಡ್ ಮಾಡಬೇಕು. ಓಕೆ, ಸೊ ವೆನ್ ಯು ಅಪ್ಲೈಡ್ ದ ಕರೆಂಟ್ DNA ಸ್ಟಾರ್ಟ್ ಮೂವ್ ಟು ಪಾಸಿಟಿವ್ ಎಂಡ್. ಓಕೆ, ಸೊ ಪಾಸಿಟಿವ್ ಎಲೆಕ್ಟ್ರೋಡ್ ಕಡೆ ಇಟ್ ಸ್ಟಾರ್ಟ್ ಮೂವ್. ದಟ್ ಪಾಸಿಟಿವ್ ಎಲೆಕ್ಟ್ರೋಡ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ ಸೊ ಆ್ಯನೋಡ್. ಸೊ ರಿಮೆಂಬರ್ ದಿಸ್, DNA move towards anode. So you should load the DNA towards cathode. Then a cathode not move towards anode. Okay, that is the property of DNA, and it is the principle behind this electrophoresis. So how the DNAs get separated? Okay, suppose if you consider the same thing, the DNA which has less kilo base pairs. So now we can divide them according to their length. The 1 kbp, 2 kbp, 3 kbp, like that, based on 10 kbp, 20 kbp, like that we can assign them. So how they are going to separate it? kbp in the kilo base pairs for a long strand of DNA we can say. So now tumba short ತಗೊಂಡಿದ್ದೀವಿ. ಇದರಲ್ಲಿ ತುಂಬಾ ಚಿಕ್ಕದಿರೋದು ಯಾವುದು ಅಂದ್ರೆ fragment number 1. So this fragment number has very short length. So when you apply the current, that fragment moves very fast. Okay, sorry, reverse not like this. So 1 kbp ತುಂಬಾ ಚಿಕ್ಕದಿದೆ 1 kbp ಅಂದ್ರೆ it move very fast. Okay, ತುಂಬಾ ಶಾರ್ಟ್ ಇರುವಂತದ್ದು ಬೇಗ ಮೂವ್ ಆಗುತ್ತೆ. So it can form the band there. Okay, and followed by the next one with according to length, the second one. So second one is settled somewhere here, then third one it forms the band somewhere here, and a fourth one, which is the larger one, it forms the band somewhere here. So like this, DNA is get separated according to their length or according to their kilo base pairs. So how many base pairs they have, based on that they are going to be separated on agarose gel. Okay, so here you cannot visualize the DNA directly, so you need some visualizer, need coloring agent. So the coloring agent used in this electrophoresis is ethidium bromide. Okay, by using ethidium bromide we are able to visualize the DNA. When you apply the ethidium bromide, actually ethidium bromide is a blue color, which gives the blue color in the electrophoresis. So when you observe under the UV light, it appears pink or orange color. Okay, so we can easily identify the fragments. Okay, so ಯಾವ ಬೇಸ್ ಪೇರ್ಸ್ಗಳಲ್ಲಿ ಅದು ಸಪರೇಟ್ ಆಗಿದೆ ಅನ್ನೋದು we can easily analyze it. Okay, so the fragment of generated by the restriction endonuclease in a chemical reaction can be separated by electrophoresis. So electrophoresis is the technique. Okay, you can separate the DNA. After the desired fragment, suppose we need this fourth fragment. So fourth fragment illide andre we can remove this and isolate it is in the purest form. ಅದನ್ನು ಎಲ್ಯೂಷನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಇದನ್ನು ಸ್ಕೂಪ್ ಮಾಡಿಕೊಂಡು ಅಲಾಂಗ್ ವಿತ್ ದ ಜೆಲ್ ಯು ಕ್ಯಾನ್ ಟೇಕನ್ ಔಟ್ ಆ್ಯಂಡ್ ಟೇಕನ್ ಇಟ್ ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಒಳಗೆ ತೊಗೊಂಡು ಅಗಾರ್ನೆಲ್ಲ ಡಿಸಾಲ್ವ್ ಮಾಡಿಸಿ ದೆನ್ ಸೆಂಟ್ರಿಫ್ಯೂಜ್ ಮಾಡಿದ್ರೆ ಅಗಾರೆಲ್ಲ ಕೆಳಗಡೆ ಸೆಟ್ಲ್ ಆಗುತ್ತೆ, ಮೇಲ್ಗಡೆ ಪ್ಯೂರೆಸ್ಟ್ ಫಾರ್ಮಲ್ಲಿ DNA ಇರುತ್ತೆ. ಸೊ ದಟ್ DNA ಕ್ಯಾನ್ ಬಿ selected for this polymerase chain reaction. That purest form DNA, ಒಂದು DNA fragment ಸಿಕ್ಕಿದೆ, it can be multiplied into billion copies. ಇದನ್ನು ಬಿಲಿಯನ್ ಕಾಪೀಸ್ ಆಗಿ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡ್ಬೋದು by using that polymerase chain reaction. Okay, yeah. So there is, after digestion, the process of cutting the DNA into many fragments is called as a digestion. After digestion, we can subject it into electrophoresis. So electrophoresis is a separation technique. Remember, it's a separation technique that can be used to separate the DNA fragments according to their length. Okay. Yeah, next move on to the next question. Question number 6. Yeah. So agarose extracted from seaweeds is used in, or just the direct question, it can be used in gel electrophoresis. Okay, so agarose adana gelidium and gracilaria. Okay, remember this is a name isolated from gelidium and gracilaria. So agar in isolate ಮಾಡ್ತೀವಿ. It's hydrocolloidal substance. That agar, highly purified agar, can be used in gel electrophoresis. Okay. Yeah. So spectrophotometry, it is used to measure the concentration in a solution. Solution ಅಲ್ಲಿ solid concentration ಎಷ್ಟಿದೆ ಅಂತ measure ಮಾಡಲಿಕ್ಕೆ we can use this photophotometry, and that absorbance, okay, absorbance of the particular ions or ions can be identified by using that spectrophotometry. So tissue culture is a technique by which we can produce 1,000 together plants in a lab in a short span of time by using an X plant. So PCR to multiply the DNA fragments. Okay, so that's it. Let's move on to the next question. Yeah. 7. In genetic engineering, the antibiotics are used. Okay, so antibiotics are ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತೀವಿ? As a selectable marker, to select healthy vector, to keep the culture free from infection, as a sequence from where replication starts. So ಯಾವುದು ಆಗಲ್ಲ, you can eliminate directly, option choose it as a selectable marker. Okay, so what is it, a selectable marker? So selectable marker is used to isolate the transformants. Okay, so transformants na isolate ಮಾಡ್ಕೊಳ್ಳೋಕ್ಕೆ we can use this antibiotic resistant gene. Okay, so ಏನು those transformants? So transformants are the bacteria which take up the rDNA. rDNA ನ ಒಳಗೆ ತಗೊಂಡಿರುವಂಥ DNA become transformants. So opposite to this one is non-transformant. Non-transformant. So this non-transformants which do not taken up the DNA. So here rDNA is present. So in this case rDNA is present. So here rDNA absent. rDNA absent. So the one which taken up that desired DNA containing plasmid become transformants. So the whole process takes place in a test tube. In a test tube we are going to add many bacteria. Of course, million together bacterial cells were there. Then we are going to add plasmids. Okay, so ಇದರ plasmids ಅನ್ನು add ಮಾಡಿರ್ತೀವಿ. Okay, so among this only few bacteria, in millions together bacteria, only few bacteria are able to take up this DNA, rDNA. So let's say, so this is the one which taken up that our desired rDNA. So this bacteria, let's say, taken up the DNA. So here also let's taken up the DNA. Okay, so see here itself 1 2 3 4 5 6 7 8 9 10. Okay, so among the 10 bacteria 1 2 3 only 3 among the 10 only 3 are transformants. And the remaining 7 are non-transformants. Okay, so if you take up this whole thing and grow in a bioreactor, so ನಮಗೆ ಬೇಕಾಗಿರೋ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ ಪ್ರಾಡಕ್ಟ್ಗಿಂತ ಬೇಡದೇ ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾನೇ ಜಾಸ್ತಿ ಬೆಳ್ಕೊಂಡ್ಬಿಡುತ್ತೆ. ಸೊ ದಟ್ ಹೋಲ್ ಬಯೋರಿಯಾಕ್ಟರ್ ಇಸ್ ಫಿಲ್ಡ್ ವಿತ್ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್. ಓಕೆ, ನಮಗೆ ಬೇಕಾಗಿರೋ ಪ್ರಾಡಕ್ಟ್ ಡಿಸೈರ್ಡ್ ಕ್ವಾಂಟಿಟಿನಲ್ಲಿ ವಿ ಆರ್ ಅನೇಬಲ್ ಟು ಒಬ್ಟೈನ್. ಸೊ ವಿ ನೀಡ್ ಟು ಸೆಲೆಕ್ಟ್ ಓನ್ಲಿ ದೀಸ್, ಬಟ್ ರಿಮೆಂಬರ್ ವಿ ಕೆನಾಟ್ ವಿಜುವಲೈಸ್ ಇಟ್. ಸೊ ಇದು ಹೇಗಾಗ್ತಾ ಇದೆ, ಯಾವ್ದು ಒಳಗೆ ತೊಗೊಂಡಿದೆ ಪ್ಲಾಸ್ಮಿಡ್ನ ಹೇಗೆ ಅಂತೇಳಿ ವಿ ಕೆನಾಟ್ ವಿಜುವಲೈಸ್. ಓಕೆ, ಸೊ ಬಟ್ ವಿ ನೀಡ್ ಟು ಐಸೋಲೇಟ್ ದಮ್. ನಿಮಗೆ ಬೇಕಾಗಿರೋದು ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್ ಮಾತ್ರ. ದೆನ್ ಹೌ ಟು taken up that transformants out. Okay, so for that purpose we are going to use this selectable marker. Okay, so if you take an example of PBR 322, in PBR 322, that plasmid of Bolivar and Rodriguez, it has 2 resistant sites, ampicillin and tetracycline. Okay, so if you cut the DNA here, okay, so ಅಲ್ಲಿ PST1 ಮತ್ತು PV1 ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಇಲ್ಲಿ ಕಟ್ ಮಾಡುತ್ತೆ. ಇಲ್ಲಿ ಕಟ್ ಮಾಡಿದ್ರೆ ವಿ ಕ್ಯಾನ್ ಇನ್ಸರ್ಟ್ ಡಿಸೈರ್ಡ್ DNA. ಲೆಟ್ಸ್ ಸೇ ಇಲ್ಲಿ ನಮಗೆ ಡಿಸೈರ್ಡ್ DNA ನಾ ಇನ್ಸರ್ಟ್ ಮಾಡಿದ್ದೇವೆ ಅಂತ ಇಟ್ಕೊಳ್ಳಿ. ನೌ ಇಟ್ ಲೂಸಸ್ ದ ಎಬಿಲಿಟಿ ಆಫ್ ಆಂಫಿಸಿಲಿನ್ ರೆಸಿಸ್ಟೆಂಟ್ ಸೈಟ್ಸ್. Okay, but it can retain the resistant against tetracycline. So if this plasmid was introduced into bacteria, remember now this bacteria can grow or chose the resistant against tetracycline. So similarly one more is there, that bacteria do not taken up this plasmid along with the normal DNA. Of course size variation ಇದೆ, ಇದು ದೊಡ್ಡದಿರುತ್ತೆ ಇದು ಚಿಕ್ಕದಿರುತ್ತೆ. Okay, so this bacteria is now transformant, whereas this bacteria is non-transformant. ಹೌದಾ? ಸೊ ನಮಗೆ ಇದು ಮಾತ್ರ ಬೇಕು, ಇದು ಬೇಡ. ಅವಾಗ ಏನು ಮಾಡ್ತೀವಿ, we are going to grow this bacteria in a media which contains tetracycline. So tetracycline containing media ಹಾಕಿದ್ರೆ, if both the bacteria were put into tetracycline containing media, the one which taken up the desired DNA ಅಂದರೆ RDNAನ ಯಾವುದು ಒಳಗೆ ತೊಗೊಂಡಿದೆಯೋ, so this is the transformant, this is non-transformant. Since it has the tetracycline resistant gene, okay, it can survive, okay, but it is unable to survive. So we are able to select the transformants. So remember in PBR322, if gene is inserted at ampicillin resistant site, we can select the transformants by growing on tetracycline. If you insert the desired gene at a tetracycline, we are able to select the transformants by growing on ampicillin. Okay, so this is the function of this selectable marker. Adana select ಮಾಡ್ಕೊಳ್ಳಿಕ್ಕೆ, transformants ಅನ್ನು select ಮಾಡ್ಕೊಳ್ಳಿಕ್ಕೆ we are going to use it. Okay, so that is the selectable marker. Okay. So different bacteria have different, plasmids have different type of selectable markers. As I already told, PUC18 has a beta galactosidase as a product, as a selectable marker, chromogenic selectable marker. Okay. So next question, that is during a heat shock to the bacterium, the temperature used for giving thermal shock is, okay, so heat shock treatment nalli so many bacteria were there, at the same time so many plasmids, recombined plasmid, let's say these are the recombined plasmids. If you mix these within a test tube and ask the bacteria to take up this plasmid, so plasmid ಒಳಗೆ ತಗೊಳ್ಳಿ ಅಂದರೆ bacteria unable to take up. Okay, so we need to forcibly move this plasmid into the bacterial cell. ನಾವೇ ಫೋರ್ಸಬ್ಲಿ ಅದನ್ನು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಒಳಗೆ ಮೂವ್ ಮಾಡೋಕ್ಕಾಗತ್ತೆ. ಓಕೆ, ಸೊ ರಿಮೆಂಬರ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹ್ಯಾಸ್ ದ ಮೆಮ್ಬ್ರೇನ್. ಓಕೆ, ಸೊ ಒಳಗಡೆ ಮೆಮ್ಬ್ರೇನ್ ಇರುತ್ತೆ. ದಿಸ್ ಮೆಮ್ಬ್ರೇನ್ ಈಸ್ ಆಸ್ ಯು ನೋ ಲಿಪಿಡ್ ಬೈಲೇಯರ್. ಇಟ್ ಈಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಲಿಪಿಡ್ ಬೈಲೇಯರ್. ಸಾರಿ, ಸೌಂಡ್ ಆಯಿತು. So lipid bilayer, this lipid bilayer has the structure like this, and remember it is lipid bilayer idu hydrophilic, idu hydrophobic, idu one property. ಜೊತೆಗೆ ಈ hydrophilic nature ಇರುವಂಥದ್ದು, it carries negative charge. Remember that DNA is also negatively charged. So let me take that membrane itself. So DNA nu kuda negative charge ಇದೆ, ಇದು negative charge ಇದೆ, ಇದು hydrophilic ಇದೆ, ಇದು ಕೂಡ hydrophilic ಇದೆ. So both are get repelled. Okay, ಅದನ್ನ ನ್ಯೂಟ್ರಲೈಸ್ ಮಾಡಬೇಕು. ಅದಕ್ಕೆ ಏನು ಮಾಡ್ತೀವಿ, we are going to add calcium chloride, CaCl2 ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ. ದಟ್ Ca+, ಇದು Ca+ ಎರಡಕ್ಕೂ ಹೋಗಿ ಬೈಂಡ್ ಆಗುತ್ತೆ, ಇಲ್ಲೂ ಬೈಂಡ್ ಆಗುತ್ತೆ, Ca+ ನ್ಯೂಟ್ರಲೈಸ್ ಆಗುತ್ತೆ. ಸೊ ಇಟ್ ಕ್ರಿಯೇಟ್ ದ ಪೋರ್ when you give the heat shock. So heat shock ಕೊಟ್ಟ ತಕ್ಷಣ pore size increase ಆಗುತ್ತೆ, so plasmid can enter easily. So this process is called as making the bacteria competent to take up the RDNA. Okay, so ಅದರಲ್ಲಿ ಕೋಲ್ಡ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಫಸ್ಟು CaCl2 ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ, ಇಮಿಡಿಯೇಟ್ಲಿ ಎಕ್ಸ್ಪೋಸ್ ಟು ಹೈ ಟೆಂಪರೇಚರ್, ದಟ್ ಈಸ್ ಹೀಟ್ ಶಾಕ್, ಇಮಿಡಿಯೇಟ್ಲಿ ದೆನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಟು ಲೋ ಟೆಂಪರೇಚರ್ ಅಗೇನ್. ಓಕೆ, ಅಲ್ಲಿ ಯಾವುದಕ್ಕೆ ಎಕ್ಸ್ಪೋಸ್ ಮಾಡ್ತೀವಿ ಅಂದರೆ 42 ಡಿಗ್ರಿ ಸೆಂಟಿಗ್ರೇಡ್ ಟು ಮೇಕ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಕಾಂಪಿಟೆಂಟ್ ಟು ಟೇಕನ್ ಅಪ್ ದ plasmid. Okay, so that is the temperature 42 degree centigrade. Let's move on to the next question. Okay, so next, which of the following enzyme is used in case of a fungus to cause a release of DNA along with other macromolecules? Okay, so here during isolation of DNA we need to use different enzymes, so depending on the cell wall material. If it is a plant cell, cell wall is cellulose. If it is a bacteria, cell wall is made up of murin or peptidoglycan. If it is a fungal hyphae, the cell wall is made up of chitin. To isolate the DNA, we need to treat them. We need to remove the cell wall first. For cellulose, we need to use the enzyme cellulase. For murin, lysozyme. And for chitin, it is chitinase. Of course, along with that, we need to use the lipase enzyme to break the lipid. Lipase and protease and RNase all are needed to isolate the DNA. So to remove this cell wall material, we need different enzymes. Okay. So lysozyme for bacteria, chitinase for fungi, cellulase for plants. So here they are asking for which of the following enzyme is used to cause a fungus. So chitinase, because the cell wall of fungi is made up of chitin. So we need to use chitinase. Okay. Yeah, that's it. Next question. So during isolation of DNA, addition of which of the following causes a precipitation of a purified DNA? So after treating with the different enzymes to remove the cell wall, we are going to subject it to centrifugation. After centrifugation, ಈಗ ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ನಲ್ಲಿ ಎಲ್ಲ ಎಂಜೈಮ್ ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿದ್ರೆ ಸೆಂಟ್ರಿಫ್ಯೂಜ್ ಮಾಡಿದಾಗ ಡೆಬ್ರೀಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸೆಟಲ್ ಹಿಯರ್, ಬಾಟಮಲ್ಲಿ ಸೆಟ್ಲ್ ಆಗುತ್ತೆ. ಲೈಕ್ ಪ್ರೋಟಿಯಸ್ ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿರ್ತಾರೆ, ಪ್ರೋಟೀನ್ ಬ್ರೇಕ್ ಡೌನ್ ಆಗಿರೋದು ಕೆಳಗಡೆ ಸೆಟ್ಲ್ ಆಗುತ್ತೆ. ದೆನ್ ಲೈಪೇಸ್ ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿರ್ತೀವಿ, ಲೈಪೇಸ್ ಎಲ್ಲ ಲಿಪಿಡ್ ಪಾರ್ಟ್ ಎಲ್ಲ ಕೆಳಗಡೆ ಬಂದು ಸೆಟ್ಲ್ ಆಗಿರುತ್ತೆ. ಕೈಟಿನೇಸ್ ಯೂಸ್ ಮಾಡಿರ್ತೀವಿ, ಅದು ಕೂಡ ಬಂದು ಸೆಟ್ಲ್ ಆಗಿರುತ್ತೆ. ಯಾವುದೇ ಆರ್ಗ್ಯಾನಿಸಮ್, ಡಿಪೆಂಡ್ ಆನ್ ಆರ್ಗ್ಯಾನಿಸಮ್. ಸೊ ಫೈನಲಿ ಈ ಪಾರ್ಟ್ ಏನಿರುತ್ತೆ ಆಫ್ಟರ್ ಸೆಂಟ್ರಿಫಿಕೇಶನ್, ದಟ್ ಲಿಕ್ವಿಡ್ ಪಾರ್ಟ್ contains the DNA, but it cannot be visualized. Small, very thin. Okay, sub microscopic ಅದು ಕಾಣಿಸೋದೇ ಇಲ್ಲ, ಅಷ್ಟು ಥಿನ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ದಟ್ ಕ್ಯಾನ್ ಬಿ, ಇದನ್ನು ಸೂಪರ್ ನಿಟೆಂಟ್ ಅಂತೀವಿ. ದಿಸ್ ಸೂಪರ್ ನಿಟೆಂಟ್ ಕ್ಯಾನ್ ಬಿ ಡಿಕ್ಯಾಂಟೆಡ್ ಇಂಟು ಸಪರೇಟ್ ಟ್ಯೂಬ್. ಈ DNAs ಇರುತ್ತೆ, ಬಟ್ ವಿ ಆರ್ ಅನೇಬಲ್ ಟು ವಿಜುವಲೈಸ್ ಇಟ್. ಟು ದಿಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಆ್ಯಡ್ ಚಿಲ್ಡ್ ಆಲ್ಕೋಹಾಲ್. ಚಿಲ್ಡ್ ಆಲ್ಕೋಹಾಲ್ ಹಾಕಿದಾಗ DNAs ಗೆಟ್ ಪ್ರೆಸಿಪಿಟೇಟೆಡ್ ಆ್ಯಂಡ್ ಯು ಕ್ಯಾನ್ ವಿಜುವಲೈಸ್ ಇಟ್. ಈ ಥರ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ, ಅದು ಕಾಣಿಸುತ್ತೆ ನಮಗೆ, ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಥ್ರೆಡ್ ಥರ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಣಿಸುತ್ತೆ. ದಟ್ ಕ್ಯಾನ್ ಬಿ ವಿಜುವಲೈಸ್. ಓಕೆ, ಸೊ ಚಿಲ್ಡ್ ಎಥನಾಲ್ ಈಸ್ ಯೂಸ್ಡ್ ಟು ಪ್ರೆಸಿಪಿಟೇಟ್ ದ DNA. ಓಕೆ, ಯಾ, ಸೊ ದಟ್ಸ್ ಇಟ್. ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್. So, which of the following is a plasmid? PBr322, BMH1, SAL1, AQR1. So remember, all these are restriction sites, are restriction enzymes. So among this vector is PBr322. So direct question. So plasmid of Bolivar and Rodriguez is PBr322. Okay, so direct question. Move on to the next question. So which of the following is a natural genetic engineer of plant? Yeast, Agrobacterium tumefaciens, E. coli and Mycoplasma. So you are familiar with this. Agrobacterium tumefaciens is the bacteria which contains a TI plasmid. That TI plasmid has the ability to transfer the desired gene. So it is Agrobacterium TI plasmid. So remember, they are going to use disarmed TI plasmid. Okay, otherwise it can cause the disease. Agrobacterium tumefaciens is able to cause the disease in plants, especially in dicot plants. It causes a gall disease called as crown gall. Okay, crown gall na cause ಮಾಡುತ್ತೆ. So when this bacteria infect the plant cell, so plant cell infect ಮಾಡ್ತಾ ಇದೆ ಅಂದರೆ it can transfer, it can enter and transfer the desired gene into plant cell. Okay, so then a genome ಒಳಗಡೆ ಸೇರಿಸುತ್ತೆ ತಗೊಂಡೋಗಿ. That's why it is called as a natural genetic engineer. Okay, so Agrobacterium tumefaciens. So the plasmid name is TI plasmid. So TI plasmid. So TI stands for tumor inducing. So tumor inducing plasmid. So in a plasmid, suppose this is the part which is able to cause the tumor in plant, so that can be removed. ಅದನ್ನು ರಿಮೂವ್ ಮಾಡಿ ಜಾಯಿನ್ ಮಾಡ್ತಾರೆ ಹಾಗೇನೆ. ಸೊ ಓಕೆ, ಅದಾದಮೇಲೆ ಅದನ್ನು ಡಿಸಾರ್ಮ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್ ಅಂತೀವಿ. ಆ ಡಿಸಾರ್ಮ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್ ಓನ್ಲಿ ಯೂಸ್ಡ್ ಫಾರ್ RDNA ಟೆಕ್ನಾಲಜಿ, ಆರ್ ಅದರ್ವೈಸ್ ಪ್ಲಾಂಟ್ ಗಾಟ್ ದ ಡಿಸೀಸ್, ಕ್ರೌನ್ ಗಾಲ್ ಡಿಸೀಸ್ ಬರೋ ಚಾನ್ಸಸ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಸೊ ಇಟ್ ಕೆನಾಟ್ ಬಿ ಯೂಸ್ ದ Agrobacterium tumefaciens ಡೈರೆಕ್ಟ್ಲಿ, ಅದರ ಪ್ಲಾಸ್ಮಿಡ್ ಡೈರೆಕ್ಟಾಗಿ ಯೂಸ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಓಕೆ, ಲೆಟ್ಸ್ ಮೂವ್ ಆನ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್. So here's a procedure through which a piece of DNA is introduced, is introduced in a host bacterium. Transduction, transformation, conjugation, replication. So ಯಾವ ಮೆಥಡ್ ಇಂದ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ genome introduce ಆಗುತ್ತೆ. ಬ್ಯಾಕ್ಟೀರಿಯಲ್ genome ಒಳಗಡೆ fragment of DNA introduced. Of course, it was experimentally proved by Griffith. Okay, R strain of bacteria transformed into S strain because of the transformation, the process called transformation, where S strain bacterial genome is entered into R strain bacteria, then convert R strain into S strain. So that process is called transformation. So transformation, conjugation and transduction. These are the three types of sexual reproduction in bacteria. Remember, in case of bacteria, there is no formation of gamete in sexual reproduction. Simply the exchange of genetic material is itself called as sexual reproduction. So here in transduction, genetic material is exchanged between two bacterial cells through bacteriophage or through virus, whereas in transformation, exchange between two bacterial cells directly. Then conjugation through conjugation bridge. Okay. So ಈಗ ಎರಡು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮಧ್ಯೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಮಧ್ಯೆ there will be a formation of conjugation bridge. So ಇಲ್ಲಿರೋ genome ಏನಾಗುತ್ತೆ, ಒಂದು ಕಡೆ replicate ಆಗುತ್ತೆ, let's say it get replicated, so this part migrated to another bacterial cell, then get joined together there. Okay, so it creates the recombination ಅಥವಾ joining ಆಗುವಂಥದ್ದು ಆಗುತ್ತೆ. So that is the three types of reproduction in bacteria, sexual reproduction. Next, move on to the next question. So which one of the following is used as a vector for cloning genes into higher organisms? Higher organisms ಗೆ ನಮಗೆ ಬೇಕಾಗಿರೋ ಜೀನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಲಿಕ್ಕೆ which one can be used? Okay, so baculovirus, no. So baculoviruses can be used as a biocontrol agents. So yesterday we discussed that. Salmonella typhimurium, no, it cannot be used. Rhizopus nigricans, no, it cannot use. So retroviruses can be used. Okay, if you insert the particular gene coding RNA into retrovirus, that RNA, if you introduce RNA, that RNA can be converted into double stranded DNA called complementary DNA, so that retrovirus can deliver that complementary DNA into host. Okay, so especially animal cells, in case of animal cell we can use retroviruses as a vectors to transfer the desired DNA. Okay, yeah. So retroviruses are the viruses which contains RNA as their genetic material, and they have the mechanism called a reverse transcription. They can convert RNA into double stranded DNA. Okay, so next question number 15. Next, which of the following are required to facilitate cloning into a vector? Okay, so gene na clone ಮಾಡಬೇಕಂದ್ರೆ ಏನೆಲ್ಲ ಇರಬೇಕು? Origin of replication, selectable marker, cloning site, all of this. Obviously each and every plasmid, if it can be used as a vector, it should have one ORI site. ORI site ಇರಬೇಕು. To select the transformant, it should have one selectable marker. ಯಾವುದಾದ್ರೂ selectable marker, antibiotic resistant gene ಇರ್ಬೋದು ಅಥವಾ chromogenic selectable marker ಕೂಡ ಇರಬೇಕಾಗತ್ತೆ. Then cloning site, it should have many cloning sites. So cloning sites are nothing but restriction sites. Restriction sites ಅಂತೀವಿ, recognition sequence ಅಂತೀವಿ, ಅಥವಾ recognition sequence ಅಥವಾ recognition site. So it's called by different names. So where restriction enzyme make a cut, ಎಲ್ಲೆಲ್ಲಿ restriction enzyme ಕಟ್ ಮಾಡೋಕ್ಕೆ ಜಾಗನ ಇರುತ್ತೋ ಆ ಜಾಗನ ನಾವು ಕ್ಲೋನಿಂಗ್ ಸೈಟ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಆಲ್ ಆಫ್ ದೀಸ್ ಇವೆಲ್ಲದೂ ಇರಬೇಕಾಗತ್ತೆ ಇಫ್ ಇಟ್ ಈಸ್ ಕನ್ಸಿಡರ್ಡ್ ಆಸ್ ಎ ಬೆಸ್ಟ್ ವೆಕ್ಟರ್. ಓಕೆ, so this thing is regarding the plasmid as a vector. So plasmids alli istu properties irbekagutte. Okay, so next move on to the next question. Yeah. So for the transformation, microparticles coated with the DNA to be bombarded with a gene gun are made up of. So it is a type of transfer of desired DNA or desired gene into plant cell. So there are two methods to make the plant a transgenic plant. One is vector method, another one is non-vector method. So into the plant cell we can deliver the desired gene by two means. One is vector method, another one is non-vector method. So vector method is by using TI plasmid of Agrobacterium tumefaciens. It's a vector method. So where TI plasmid is act as a vector. In another method where vector is not going to be used, okay, non-vector method, so it's the direct method by using gene gun. Okay, or it is also called as biolistic method. So in this method, so there is a gun like structure. Of course ನಾರ್ಮಲ್ ಗನ್ ಥರ ಅಲ್ಲ, ಇದು ಸ್ವಲ್ಪ ಡಿಫ್ರೆಂಟಾಗಿರುತ್ತೆ. ಸೊ ಇದು ಜೀನ್ ಗನ್ ಇರುತ್ತೆ. ಸೊ ಈ ಜೀನ್ ಗನ್ ಒಳಗಡೆಗೆ we can introduce some particles, very minute particles. Okay, so these minute particles could be, these minute particles can be loaded with a desired gene. Idr melgade desired gene na coat ಮಾಡಿ we can load here and it can be transferred to a plant cell. So one particular speed na calculate ಮಾಡಿಕೊಂಡು they are going to trigger it. So this particle go and bind to any one of the cell. So remember ಎಲ್ಲ ಸೆಲ್ಸ್ಗೆ ಹೋಗಿರಲ್ಲ. ಸೊ ಇಲ್ಲಿ ಮಾರ್ಕ್ ಆಗಿರುತ್ತೋ ಅಂಥ ಸೆಲ್ಸನ್ನು ದೇ ಟೇಕನ್ ಔಟ್, ಈ ಪಾರ್ಟನ್ನು ಹೊರಗೆ ತೊಗೋತಾರೆ. ಸೊ ತೊಗೊಂಡು ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಲ್ಚರ್ಡ್ ಇನ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್. ಸೊ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ನಲ್ಲಿ ಗ್ರೋ ಮಾಡ್ತಾರೆ. ಸೊ ಗ್ರೋ ಮಾಡಿ ಪ್ಲಾಂಟ್ಲೆಟ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಪ್ರೊಡ್ಯೂಸ್ಡ್. ಅದರಲ್ಲಿ ಯಾವ ಪ್ಲಾಂಟ್ಲೆಟಲ್ಲಿ ನಮ್ಗೆ ಬೇಕಾಗಿರೋ ಜೀನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿದೆ, ಲೇಟರ್ ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಫೈಂಡ್ ಇಟ್. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ದ ಜೀನ್ ಗನ್ ಮೆಥಡ್. ಸೊ ಹಿಯರ್ ದೀಸ್ ಪಾರ್ಟಿಕಲ್ಸ್ which are coated with the DNA are made up of either gold or tungsten. ಎರಡಿಂದ ಆಗಿರುತ್ತೆ. ಓಕೆ, ನಾಟ್ ಸಿಲ್ವರ್ ಆರ್ ಪ್ಲಾಟಿನಮ್, ಯಾವುದು ಅಲ್ಲ, ಗೋಲ್ಡ್ ಮತ್ತು ಟಂಗ್ಸ್ಟನ್ ಪಾರ್ಟಿಕಲ್ಸಿಂದ ಆಗಿರುತ್ತೆ. ಓಕೆ, ಯಾ. ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ vectorless method to transfer the desired DNA. Okay, so next question. Yeah. So which one of the following palindromic base sequence in DNA can be easily cut at about the middle by some particular restriction enzyme? Okay, so ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮು ಎಲ್ಲಿ ಕಟ್ ಮಾಡುತ್ತೆ, ಯಾವುದು ಪ್ಯಾಲಿಂಡ್ರೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಆಗಿ ಆ್ಯಕ್ಟ್ ಆಗುತ್ತೆ? ಸೊ ಲೆಟ್ ಸಿ, ಸಿ ಜಿ ಟಿ ಟಿ ಸಿ ಜಿ ನೋಡಿ, it should be read same in both the direction, so it can't read it. GATATG, GATATG, GA, no, it is not a palindrome. GAATTC, GAATTC. Okay, so actually it should be moved further. ಇದು ನೇರವಾಗಿ ಬರಬೇಕಿತ್ತು. ಸೊ ಇಟ್ ಕ್ಯಾನ್ ರೀಡ್ ಸೇಮ್. CACGTA, CACGTA ಇಲ್ಲ. ಸೊ ದಿಸ್ ಇಸ್ ದ ಓನ್ಲಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್. restriction or palindromic sequence or recognition sequence or restriction sequence where it made a cut between G and A. So this GAATTC is a recognition sequence for Echo R1 Escherichia coli strain R enzyme number 1. Echo R1 restriction sequence is GAATTC. Okay, so remember this. So next, the first restriction endonuclease reported was, as already discussed, it is HIND2. Okay, so HIND2 is the first restriction enzyme reported. So Haemophilus influenzae. Okay, yeah, Escherichia coli, then Bacillus amyloliquefaciens strain H, and enzyme number 1, BMH1. So let's move on to the next question. Yeah. So in agarose space, in agarose gel electrophoresis, DNA migrates towards the negative electrode. No, as already told, DNA move towards positive electrode because DNA being a negative, it should move towards the positive end. Then supercoiled plasmids, supercoiled, migrates slower than their nicked counterparts. Okay, so supercoiled plasmids migrate slower than their nicked counterpart. Nicked ಅಂದ್ರೆ ಕಟ್ ಮಾಡಿರುವಂಥದ್ದು. Then larger molecules migrate faster than smaller molecules. Okay, then ethidium bromide can be used to visualize the DNA. Okay, so supercoiled plasmids migrate slower than nicked counterparts. It is not the right one. So larger molecules migrate faster than smaller molecules. It is also not. Then ethidium bromide can be used to visualize the DNA. So this is the right one. So ethidium bromide is a strain used to visualize the DNA during electrophoresis. It is actually, ethidium bromide is blue color. When you visualize it under UV light, it appears pink or orange or little red color. Okay. Yeah, so that's it. So next question. Yeah. So the polymerase enzyme used in PCR, the DNA polymerase enzyme used in PCR is the DNA polymerase 1. No, because in PCR it should tolerate high temperature up to 94 degrees centigrade, because in denaturation we are going to heat up the DNA up to 94 degrees centigrade. ಅಷ್ಟು ಎಂಜೈಮ್ ತಡ್ಕೊಳ್ಳೋ ಶಕ್ತಿ ಇರಬೇಕು. So it can tolerate the stack polymerase. The reverse transcriptase no, then a restriction endonuclease no. Okay, so TAC polymerase is the enzyme which can 4 degrees centigrade. So TAC, the name TAC originated from Thermus aquaticus. Okay, so it is Thermus aquaticus. So Thermus aquaticus anta andre it is a bacteria, it is archaebacteria which lives in aquaticus. Thermus andre high temperature, high water. Aquatic andre hot springs anta ಏನು ಹೇಳ್ತೀವಿ. In hot springs this Thermus aquaticus is there. Since in hot spring this bacteria replicated under the high temperature. Okay, so boiling temperature alli bacteria replicate ಆಗ್ತಾ ಇದೆ ಅಂದ್ರೆ that enzyme has the ability to tolerate high temperature also. So that is the reason why we are going to isolate this polymerase enzyme from this Thermus aquaticus and use it in PCR. That is the reason why it is named as Thermus aquaticus. Thermus aquaticus called as Taq polymerase. Okay, so Taq polymerase enzyme. Okay, let's move to the next question. Yeah, so the first step in the PCR is denaturation, primer extension, annealing and cooling. Okay, so in any PCR, so what happens in the PCR? So let me explain simply. So it has the complementary base pairing or double stranded DNA. When you heat this to 94 degrees centigrade, the first thing they are going to be separated. So ee reeti separate agtave, strands are get separated. So yak separate agutte andre when you subject to 94 degrees centigrade the hydrogen bond breaks. Hydrogen bond break agi they separate agutte. So to this separated strands, ee separate agiro strands na ನಾವು multiply ಮಾಡ್ತೀವಿ by using enzyme, the Taq polymerase. Okay, so after this separation ಆದ ನಂತರ initiate ಆಗಬೇಕು DNA, replication should be initiated. So we are going to lower the temperature to 40 to 50 degrees centigrade. 40 ಇಂದ 50 degree centigrade ತನಕ we are going to lower the temperature. So ಈ ರೀತಿ ಲೋ ಮಾಡಿದಾಗ ಈ process ಏನಿದೆ that is called annealing. So during annealing primers were added, forward primer and reverse primer were added on both the strands at annealing temperature. Okay, so after annealing when the primers were added the temperature is again raised to idana temperature na rise ಮಾಡ್ತಾರೆ 60 to 70 degree centigrade. Okay, so ee temperature na rise ಮಾಡೋ process ಏನಿದೆ this is called extension temperature. So during extension DNA polymerase enzyme go on add the respective nucleotides. ee ರೀತಿ DNA na synthesize ಮಾಡುತ್ತೆ after the polymerase enzyme. Okay, so like this new strand of DNA is going to be synthesized. So first step is denaturation, second step is annealing, third step is primer extension. So remember the temperatures. So it is 94 degrees centigrade, then annealing temperature 40 to 50 degrees centigrade, then primer extension ಆಗಬೇಕು ಅಂತಂದರೆ it should be subjected to 60 to 70 degree centigrade. Okay, so then a particular timings na fix ಮಾಡಿರ್ತಾರೆ in a PCR machine, it go on adding the Taq, of course Taq polymerase add ಮಾಡಿರ್ತೀವಿ, so it go on add the respective nucleotides. So the nucleotides which are used here it called as oligonucleotides, ಅಂದರೆ ಆರ್ಟಿಫಿಷಿಯಲಿ ಸಿಂಥಸೈಸ್ ಮಾಡಿರ್ತಾರೆ. ಸೊ ದೇ ಹ್ಯಾವ್ ಕಾಲ್ಡ್ DNTPs. ಓಕೆ, ಅದನ್ನು DNTPs ಅಂತ ಹೇಳ್ತೀವಿ, ಡಿಯಾಕ್ಸಿ ರೈಬೋ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಟ್ರೈಫಾಸ್ಪೇಟ್. ಇದರಲ್ಲಿ ಟ್ರೈಫಾಸ್ಪೇಟ್ಸ್ ಇರುತ್ತೆ. ಸಪೋಸ್ ಇಟ್ ಈಸ್ ಅಡಿನಿನ್ ಅಂದರೆ ಅದರಲ್ಲಿ ಟ್ರೈಫಾಸ್ಪೇಟ್ ಇರುತ್ತೆ. ಇದರಲ್ಲಿ 2 ಫಾಸ್ಪೇಟ್ ಬ್ರೇಕ್ ಆಗಿ ದೇ ರಿಲೀಸ್ ದ ಎನರ್ಜಿ, ದಟ್ ಎನರ್ಜಿ ಇಸ್ ಯುಟಿಲೈಸ್ಡ್ ಟು ಕ್ರಿಯೇಟ್ ದ ಬಾಂಡ್, ಟು ಕ್ರಿಯೇಟ್ ದ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಚೈನ್. ಓಕೆ, ಸೊ DNTPs were going to be used in PCR. Okay, so next move to the next question. So for a transformation with recombinant DNA, the bacterial cell must first be made competent, which means, what is the meaning of made the bacterial cell competent? So should increase their metabolic reaction, should decrease their metabolic reaction, increase the efficiency with which DNA enters the bacterium, the ability to dry fast. Okay, so divide fast, sorry, idu agalla, idu agalla, idu agalla. Yakendre namige illi transformation antandre we need to deliver our desired plasmid RNA into bacteria to make that bacteria to efficiency, to increase the efficiency of a bacterial cell to take care of the rDNA. That is called as made the bacterial cell competent. Okay, so how to made it? By heat shock treatment. So first into the test tube we are going to add the bacterial cell plasmids and the calcium chloride subjected to low temperature treatment. Then during low temperature treatment, calcium make the neutralized. DNA can be neutralized by the divalent cation. So later immediately transfer into 42 degrees centigrade. Then it create the pores, increases the pore size in the membrane. So plasmid can enter inside. Then immediately subjected to low temperature treatment, calcium will be released from the DNA. So DNA is get transferred. So that is the method we are going to use in that transformation. Okay. So next, move to the next question. Yeah. So after completion of biosynthetic stage, the product be subjected through a series of process before it is to marketing as a finished product. This series of process is called, okay, upstream, downstream, elution, insertion, inactivation. So in a bioreactor, after selecting the transformants select madadmele, of course by using selectable marker we are going to select them. Suppose this is a bioreactor, single stirred bioreactor. So we are going to add the bacterial cell, ಯಾವುದು DNA ತಗೊಂಡಿರುತ್ತೋ ಟ್ರಾನ್ಸ್ಫರ್ಮೆಂಟ್ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆ್ಯಡ್ ಮಾಡಿದ್ರೆ, ಸೊ ಇಟ್ ಅಂಡರ್ಗೋ ರ್ಯಾಪಿಡ್ ಮಲ್ಟಿಪ್ಲಿಕೇಶನ್ ವಿದಿನ್ ದ ಬಯೋರಿಯಾಕ್ಟರ್ ಅಂಡ್ ಪ್ರೊಡ್ಯೂಸಸ್ ದ ಪ್ರಾಡಕ್ಟ್, and that product is present within this media. So we can decant that media, ಆ ಮೀಡಿಯಾನ ಡಿಕ್ಯಾಂಟ್ ಮಾಡಿಕೊಂಡು ಮತ್ತೆ ಫ್ರೆಶ್ ಬ್ಯಾಚನ್ನು ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಬ್ಯಾಚ್ ವೈಸ್. ಓಕೆ, ಅದನ್ನು ಬ್ಯಾಚ್ ನಂಬರ್ಸ್ ಅಂತ ಕೊಡ್ತಾರೆ ಅದಕ್ಕೆ. ದೆನ್ ಪ್ರಾಡಕ್ಟ್ ಏನಿರುತ್ತೆ, ದಟ್ ಪ್ರಾಡಕ್ಟ್ ಕ್ಯಾನ್ ಬಿ by using different separation technique we can separate the product. ಆ ಪ್ರಾಡಕ್ಟನ್ನು ಅಷ್ಟೇ ಸಪ್ರೇಟ್ ಮಾಡ್ತೀವಿ. ಓಕೆ, ಸೊ ಈಗ 1000 ಲೀಟರ್ ಒಳಗಡೆ ಮೀಡಿಯಾ ಒಳಗಡೆ ಲೆಟ್ಸ್ ಎ ಸಿಂಪಲ್ ಟರ್ಮಲ್ಲಿ ಒಂದು 500 ಗ್ರಾಮ್ಸ್ ಮಾತ್ರ ಪ್ರಾಡಕ್ಟ್ ಇದೆ ಅಂದರೆ ಯು ನೀಡ್ ಟು ರಿಮೂವ್ ದಟ್. ಅಲ್ಲಿ ವಾಟರ್ನೆಲ್ಲ ಅಥವಾ ಮೀಡಿಯಾ ರಿಮೂವ್ ಮಾಡಿ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸಪ್ರೇಟ್ ದ ಪ್ರಾಡಕ್ಟ್. ಆ ಪ್ರಾಡಕ್ಟ್ನ ಸಪ್ರೇಟ್ ಮಾಡಿ ಅದಕ್ಕೆ ಬೇಕಾಗಿರೋ ಸೂಟೇಬಲ್ ಪ್ರಿಸರ್ವೇಟಿವ್ಸ್ ಆ್ಯಡ್ ಮಾಡಿ ಕ್ಲಿನಿಕಲ್ ಟೆಸ್ಟನ್ನ ಮಾಡಿ, if it is a drug clinical trials ಎಲ್ಲ ನೋಡಿ ಫೈನಲಿ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ರಿಲೀಸ್ ಇಟ್ ಇನ್ ದ ಮಾರ್ಕೆಟ್. ಸೊ ಈ ಪ್ರೋಸೆಸ್ ಏನಿದೆ ಈ ಬಯೋರಿಯಾಕ್ಟ್ರಲ್ಲಿ ಅದು ಮುಗಿದ ನಂತರ ಈ ಪ್ಯೂರಿಫಿಕೇಷನ್, ಐಸೋಲೇಷನ್, ಪ್ಯೂರಿಫಿಕೇಷನ್ ಆ್ಯಂಡ್ ಟೆಸ್ಟಿಂಗ್ ಆ್ಯಂಡ್ ರಿಲೀಸಿಂಗ್ ಏನಿದೆ, ಸೊ ದಟ್ ಪ್ರೋಸೆಸ್, ದ ಹೋಲ್ ಪ್ರೋಸೆಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಡೌನ್ ಸ್ಟ್ರೀಮ್ ಪ್ರೋಸೆಸ್. ಓಕೆ, ಸೊ ಇನ್ ಡೌನ್ ಸ್ಟ್ರೀಮ್ ಪ್ರೋಸೆಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಪ್ಯೂರಿಫೈ ಇಟ್. ಸೊ ಪ್ಯೂರಿಫೈ ಮಾಡಿ ಫೈನಲ್ ಪ್ರಾಡಕ್ಟನ್ನು we are going to release it into the market. Okay, so that is the process. Yeah. So let's move to the next process. Yeah, so stirrer tank bioreactors have been designed for addition of preservatives to the product. Remember, preservative is not going to add to the bioreactor. So purification of the product, of course it is the purification of the product could be done after the product is going to be produced in the bioreactor. So ಅದಕ್ಕೆ ಡಿಸೈನ್ ಮಾಡಿಲ್ಲ. ಎನ್ಶೂರಿಂಗ್ ಅನೈರೋಬಿಕ್ ಕಂಡೀಷನ್ ಇನ್ ದ ಕಲ್ಚರ್. ಆಫ್ ಕೋರ್ಸ್ ಇಫ್ ದ ಪ್ರಾಡಕ್ಟ್ ಈಸ್ ಅನೈರೋಬಿಕ್ ಪ್ರಾಡಕ್ಟ್ ಲೈಕ್ ಆಲ್ಕೋಹಾಲ್, ವಿ ನೀಡ್ ದಿಸ್ ಅನೈರೋಬಿಕ್ ಕಂಡೀಷನ್. ಯಾಕೆ ಅಂದರೆ ಆಲ್ಕೋಹಾಲ್ ಪ್ರೊಡಕ್ಷನ್ಗೆ ಅನೈರೋಬಿಕ್ ಕಂಡೀಷನ್ ಬೇಕು. ಇಫ್ ಇಟ್ ಇಸ್ ಏರೋಬಿಕ್ ಕಂಡೀಷನ್, ಆಲ್ಕೋಹಾಲ್ ಈಸ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಪ್ರೊಡ್ಯೂಸ್. ದೆನ್ ಅನೈರೋಬಿಕ್ ಕಂಡೀಷನ್ ಮೇಂಟೈನ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ ಅಂದರೆ ಇಟ್ ಈಸ್ ನಾಟ್ ಎ ಬಯೋರಿಯಾಕ್ಟರ್, ಇಟ್ ಬಿಕಮ್ ಫರ್ಮೆಂಟರ್. So fermenter ಅಂತಂದರೆ this is the condition, we need to maintain anaerobic condition, but we are talking about bioreactor. In bioreactor we are going to use the aerobic bacteria. Okay, so aerobic bacteria ಅಂದರೆ oxygen supply should be there, it should be aerobic condition. So availability of oxygen throughout the process. ಆ ಪ್ರೋಸೆಸ್ನ ಆಕ್ಸಿಜನ್ ಕಂಟಿನ್ಯೂಸ್ ಸಪ್ಲೈ ಇರೋ ಥರ ನೋಡ್ಕೊಳ್ಳೋದು ಯಾವುದು ಅಂತ ಅಂದರೆ ದಟ್ ಸ್ಟಿರರ್ ಟ್ಯಾಂಕ್ನಲ್ಲಿರುವಂಥ ದಟ್ ಸೆಂಟರ್ ಒನ್. ಏನು ಫ್ಯಾನ್ ಥರ ಏನಿರುತ್ತೆ ದಿಸ್, ಓಕೆ, ರೊಟೇಟ್ ಆಗ್ತಾನೇ ಇರುತ್ತೆ, ಗ್ರ್ಯಾಜುವಲಾಗಿ ರೊಟೇಟ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ದಟ್ ಮೇಂಟೈನ್ ದ ಯೂನಿಫಾರ್ಮಿಟಿ ಆಫ್ ಆಕ್ಸಿಜನ್ ಸಪ್ಲೈ ಟು ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ವಿಚ್ ಆರ್ ಗ್ರೋಯಿಂಗ್ ಇನ್ ದಟ್ ಮೀಡಿಯಾ. ಓಕೆ, ಇನ್ ಅ ಸ್ಪಾರ್ಜರ್ ಸ್ಟಿರರ್ ಟ್ಯಾಂಕ್ ಆಲ್ಸೋ. ಸೊ ಒನ್ ಮೋರ್ ಟೈಪ್ ಇಸ್ ದೇರ್, ದಟ್ ಸ್ಪಾರ್ಜರ್ ಸ್ಟಿರರ್. ಥ್ರೂ ಎ ಸ್ಪಾರ್ಜರ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಜನರೇಟ್ ದ ಬಬಲ್ಸ್, ಆಕ್ಸಿಜನ್ ಬಬಲ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಸೊ ಇಟ್ ಹ್ಯಾಸ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಲೈಕ್ ದಿಸ್. ಸೊ ದಿಸ್ ಇನ್ಕ್ರೀಸಸ್ ದ ಸರ್ಫೇಸ್ ಏರಿಯಾ. Okay, so oxygen supply continuous provide ಆಗ್ತಾ ಇರುತ್ತೆ ಇಲ್ಲಿ. Okay, so in case of sparged stirrer tank, so in both the case it was maintaining oxygen supply, nutrient supply, optimum temperature, optimum pH, ಅದೆಲ್ಲದನ್ನು ಮೇಂಟೈನ್ ಮಾಡ್ತಾ ಇರ್ತೀವಿ in case of bioreactors to ensure that bacteria can grow happily and produce the product. Okay, so that's it. Okay, let's move to the next question. Yeah, So which of the following statement is incorrect regarding the PCR? In PCR, 2 primers are used. Yes, we are going to use the 2 primers. One is a forward primer, another one is a reverse primer. So DNA replication ಆಗಬೇಕಂದ್ರೆ during annealing, when it reduce the temperature, ಈ ಕಡೆಗೆ ಒಂದಿಷ್ಟು ಆಲಿಗೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಆ್ಯಡ್ ಆಗುತ್ತೆ, ಈ ಕಡೆಗೆ ಒಂದಿಷ್ಟು ಆಲಿಗೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಆ್ಯಡ್ ಆಗುತ್ತೆ. ಸೊ ದಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ಇಟ್ ಮೂವ್ ಇನ್ ದಿಸ್ ಡೈರೆಕ್ಷನ್, ಸೊ ಕಾಲ್ಡ್ ಫಾರ್ವರ್ಡ್. ಸೊ ದಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ಫಾರ್ವರ್ಡ್ ಪ್ರೈಮರ್ ಆ್ಯಂಡ್ ಇದು ಈ ಆರ್ಡ್ರಲ್ಲಿ ಬರುತ್ತೆ. ಸೊ ದಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ರಿವರ್ಸ್ ಪ್ರೈಮರ್. ಸೊ ಯಾಕೆ ಅದು ಫಾರ್ವರ್ಡ್ ಮತ್ತು ರಿವರ್ಸ್ ಪ್ರೈಮರ್ ಬೇಕಾಗುತ್ತೆ ಅಂದರೆ, ಸೊ ಇಟ್ ಶುಡ್ ಬಿ ಬಿಗಿನ್ ವಿತ್ 5' ಎಂಡ್ ಅಲ್ವಾ. ಸೊ 5' ಅಂದರೆ ಇಲ್ಲಿ 3 ಇರುತ್ತೆ, ಇಲ್ಲಿ 5, ಇಲ್ಲಿ 5, ಇಲ್ಲಿ 3. ಸೊ ಇಲ್ಲಿಂದ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ಈ ಕಡೆಯಿಂದ ಸ್ಟಾರ್ಟ್ ಆಗ್ತಾ ಇದೆ ಅಂದರೆ ಒಂದು ಫಾರ್ವರ್ಡ್ ಡೈರೆಕ್ಷನ್ಗೆ ಹೋಗುತ್ತೆ, ಇನ್ನೊಂದು ರಿವರ್ಸ್ ಡೈರೆಕ್ಷನಲ್ಲಿ ಬರುತ್ತೆ. ಸೊ ಕಾಲ್ಡ್ ಫಾರ್ವರ್ಡ್ ಪ್ರೈಮರ್ ಆ್ಯಂಡ್ ರಿವರ್ಸ್ ಪ್ರೈಮರ್. ಸೊ ದೇಮ್ ಸೇಮ್ ಕೈಂಡ್ ಆಫ್ ಪ್ರೈಮರ್ಸ್ ವರ್ ಆಲ್ಸೋ ಆ್ಯಡೆಡ್ ಡ್ಯೂರಿಂಗ್ DNA replication, ಬಟ್ ದೋಸ್ ಪ್ರೈಮರ್ಸ್ ವರ್ ಆ್ಯಡೆಡ್ ಬೈ ಪ್ರೈಮೇಸ್ ಎನ್ಸೈಮ್. ಅಲ್ಲಿ RNA ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಆ್ಯಡ್ ಆಗುತ್ತೆ ಫಸ್ಟಲ್ಲಿ. ಓಕೆ, ದೆನ್ DNA nucleotides add ಆಗುತ್ತೆ. ಡ್ಯೂರಿಂಗ್ ಪ್ರೂಫ್ ರೀಡಿಂಗ್ ದಟ್ RNA nucleotides were removed and DNA nucleotides were added in case of DNA replication, but here directly DNA primers were added. So based on their complementarity they are come and bind there. Okay, so exactly ಇಲ್ಲೇ ಬೈಂಡ್ ಆಗಬೇಕಂತೇನಿಲ್ಲ, ಬೇಕಾದ್ರೆ ಮುಂದಕ್ಕೂ ಬೈಂಡ್ ಆಗಿರಬಹುದು ಡಿಪೆಂಡಿಂಗ್ ಆನ್ ದ ಕಾಂಪ್ಲಿಮೆಂಟಾರಿಟಿ. ಅದು ಎಲ್ಲಿ ಕಾಂಪ್ಲಿಮೆಂಟಾರಿಟಿ ಇದೆ ಅಲ್ಲಿಗೆ ಹೋಗಿ ಬೈಂಡ್ ಆಗುತ್ತೆ, ಅಲ್ಲಿಂದ ಮುಂದಕ್ಕೆ DNA replication can occur. So before anything we go with the PCR, we should know the sequence. A particular fragment of DNA ಇದು ಸ್ಟಾರ್ಟಿಂಗ್ ಸೀಕ್ವೆನ್ಸು ಮತ್ತು ಇಲ್ಲಿ ಎಂಡ್ ಸೀಕ್ವೆನ್ಸ್ ಎರಡು ಗೊತ್ತಿರಬೇಕಾಗತ್ತೆ. ದೆನ್ ಓನ್ಲಿ ವಿ ಆರ್ ಏಬಲ್ ಟು ಡಿಸೈನ್ ದ ಆಲಿಗೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್. ಅಂದರೆ ಪ್ರೈಮರ್ಸನ್ನು ಡಿಸೈನ್ ಮಾಡಬೇಕಂದ್ರೆ ವಿ ಶುಡ್ ನೋ ದ ಸೀಕ್ವೆನ್ಸ್ ಇನಿಷಿಯಲಿ. ಓಕೆ, ಅಂದರೆ ಅದನ್ನು ಸೀಕ್ವೆನ್ಸ್ ನೋಡಿಕೊಂಡೇ ಅದನ್ನು ಸಿಂಥಸೈಸ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಸೊ ಟ್ಯಾಕ್ DNA ಪಾಲಿಮರೇಸ್ ಈಸ್ ರಿಲೇಟೆಡ್ ಫಾರ್ PCR. ಓಕೆ, tag polymerase enzyme is used in PCR to multiply the DNA, amplify the DNA. Tag DNA polymerase is not thermostable. No, should be thermostable. It should have the ability to tolerate that high temperature. Like during PCR, we are going to increase the temperature up to 94 degrees centigrade to make the double stranded DNA into single strand. And denaturation ಮಾಡಬೇಕಂದ್ರೆ 94 degrees centigrade. So remember, ನಾವೇನು ಮಾಡ್ತೀವಿ, ಈ ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಒಳಗಡೆ DNA ಹಾಕಿರ್ತೀವಿ, double stranded DNA. We are going to add primers. So primers add ಮಾಡಿರ್ತೀವಿ, DNTPs add ಮಾಡಿರ್ತೀವಿ. Then we are going to add polymerase enzyme. ಎಲ್ಲದೂ ಇದರೊಳಗೆ ಇರುತ್ತೆ. When the test tube is heated to 94 degrees centigrade in PCR machine, okay, normal polymerase enzyme ಆದರೆ it get denatured, ಆ ಟೆಂಪ್ರೇಚರ್ ತಡ್ಕೊಳ್ಳಲ್ಲ. That is the reason why we are going to add tag polymerase. Okay, so multiple copies of gene can be synthesized in PCR. Okay, so multiple copies ಅಂದರೆ after 30 to 40 cycles we are able to get 1 billion copies of DNA. So this is also right. So the wrong one is this one. Okay, so tag DNA polymerase is thermostable. It should be thermostable. Okay. So let's go to the next question. So which of the following statement is incorrect? So ECOR1 cut the DNA between bases G and A. Of course, you are familiar with that GAATTC is a palindromic sequence. It make a cut between G and A. So that is right. Then each ECOR1 restriction endonuclease are specific palindromic nucleotide sequence to cut it. Okay, so adike specific palindromic sequence ಬೇಕಾಗುತ್ತೆ. That is also right. When cut by the same restriction enzyme, the fragments do not have the same kind of sticky ends. No. When you use the same restriction enzyme to cut the plasmid and the isolation of a desired DNA, so both have the sticky ends. Okay, then only that we can join the DNA. Okay, so making multiple identical copies of any DNA is called as cloning. Yes, it is right. Okay, so the wrong option, incorrect one is, if you use the same restriction enzyme, it should create the same kind of sticky ends, which should be complementary. Okay, the sticky ends na produce ಮಾಡುತ್ತೆ. Okay, next move on to the next question. Yeah, so which of the following is not correct about PBR322? It was constructed by using DNA derived occurring plasmids of E. coli. Okay, so this PBR322, it was constructed by using the plasmid of Bolivar and Rodriguez. Bolivar and Rodriguez isolated the one plasmid from E. coli bacteria and they stitched different restriction sites and specific selectable markers that ampicillin tetracycline resistant genes were introduced into that E. coli plasmid. So the name is PBR322. It has the 2 drug resistant genes, tetracycline resistant and ampicillin resistant. Of course, both are right. Then it was developed by Bolivar and Rodriguez. Yes, it is right. So selectable markers present in it recombinants can recombinants from non-recombinants and their inability to produce color in the chromogenic substrate. Okay, so remember this ampicillin and tetracycline, they are not chromogenic. They are not chromogenic selectable marker. Instead, they are antibiotic resistant selectable marker. So this chromogenic selectable marker example PUC18. Okay, so not this. So this is the correct. Okay, so it is not correct about the PBr322. Okay, so next, move to the next question. Which of the following statements are incorrect? So genetic engineering is also called as recombinant DNA technology. Of course, it is called as recombinant DNA technology by combining the genes of two different organisms. So to the one organism, you can introduce alien DNA, that is called a recombinant DNA technology, and it is also called as genetic engineering. So genetic engineering is nothing but altering the genome of an organism. That is genetic engineering. Okay. So bacteriophages is not used as a vector. No, bacteriophage can be used as a vector. Okay. So bacteriophage can be used as a vector. Okay, yeah. So to transfer the desired gene into bacteria, we can use it. Okay. So Malayalam is a palindrome sequence. If you read in any direction, it reads the same. Okay, so it is right. So ethidium bromide cannot be used for styling DNA. No, ethidium bromide is the style used to visualize the DNA during electrophoresis. Okay. So 2 and 4 are the right option. Okay. So let's move to the next question. Yeah. So statement 1, in a recombinant DNA technology, human genes are often transferred into bacteria. So it's a prokaryote or yeast eukaryote. Okay, so that is AC and BAC. You might remember that AC and BAC. So AC stands for yeast artificial chromosome, BAC bacterial artificial chromosome. We can isolate the DNA or a specific gene, and it can be introduced into a yeast genome or bacterial genome. Okay, that can be preserved in the gene bank as well. Okay, so statement 1 is right. So statement 1 is right. So statement 2, both bacteria and yeast multiply very fast to form a huge population which express the desired gene. Of course, so both have the ability to consider the foreign DNA which is introduced there into themselves, and they multiply along with that foreign gene also. So of course both the statements are right. Along with that, let's see the option first. So statement 1 and statement 2 are true, and statement 2 is the correct explanation for the statement 1. Yeah, yake ನಾವು ಅದಕ್ಕೆ ಇನ್ಸರ್ಟ್ ಮಾಡ್ತೀವಿ ಅಂದರೆ, so both bacteria and yeast multiply very fast to form a huge population which express the desired gene. ನಾವು ಇನ್ಸರ್ಟ್ ಮಾಡಿರೋ desired gene express ಆಗುತ್ತೆ. That is the reason why we are going to insert into bacteria and yeast. So statement is right. Okay. Yeah. So 29. So that's it. Yeah. Next, move to the next question. Let's move to the next question. Question number 30. Okay. So match the column 1 with the column 2 and select the correct answer using the codes given below. Okay. So in column 1, plasmid, AMP, TI plasmid, chitinase. So selectable marker, extra chromosomal DNA, enzyme, Agrobacterium tumefaciens. So there should be space. So let's see. So plasmid. So what it is? It is whether it is a selectable marker or extra chromosomal DNA or enzyme or Agrobacterium tumefaciens. So plasmid is a extra chromosomal DNA. So it is present in bacteria. Okay. So sometimes the bacteria have many plasmids of the same kind because of the auto-replication. So because plasmid has their own oocyte, it can replicate on its own. Okay, without depend on the bacterial genome. AMP, it is ampicillin. So what is that? Ampicillin is a selectable marker. So TI plasmid could be isolated from Agrobacterium tumefaciens. So chitinase is an enzyme used during isolation of DNA from fungi. Okay, so plasmid A2. So search for A2, A2. So by looking into this one only, you can choose the option. So A2, B1, B1, C4, C4, D3, D3. So it is right. So this is the right option. move to the next question. Okay, so match the item. Let's match the following again: recombinant DNA technology, cloning vehicle, macromolecular separation, DNA ligase, then vector, sealing enzyme, electrophoresis, genetic engineering. Okay, so recombinant DNA technology is also called as, okay, so here is the one. It is nothing but genetic engineering. That means A4. So search for A4. So 2 were there. So you can eliminate it. If you know very well, you can eliminate the 2. Sorry. A4 is the answer. It should be retyped. This can be eliminated. Okay. So cloning vehicles, which are used as a cloning vehicles. So cloning vehicle is vector. So B1. So in both the case it is B1, you cannot eliminate now. Macromolecular separation. So separation can be done by electrophoresis. C3. Okay, so C3. Here is the C3. Then it is not a wrong. It's a wrong thing. DNA ligase is a sealing enzyme. We can join the DNA. So it is D2. So it is sorry. It is not H. It should be D2. Okay. So it should be eliminated, and the correct one is this. Okay. So hope you got it. Yeah, next move to the next question. Again match the following: echo 1 BAMH1, that is BAMH1, hint 3 PBR322. So what are these? Okay, so echo R1 Escherichia coli, that is A3. So it is A3. So only one is there. So no doubt you can select it as an answer. So let me check it once. Confirm or not. BAMH1 Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. B1. That's right. Hint 3 Haemophilus influenzae. Okay. So that is C2. PBR322. It is an artificial plasmid or a tailored plasmid produced by Bolivar and Rodriguez. Okay. So PBR322 base plasmid is from E. coli. Okay. So that's right. Next question. Let's move on to the next question. So which of the following is incorrectly matched? Okay, so ಯಾವುದು incorrect ಆಗಿ match ಆಗಿದೆ ನೋಡಿ. Agarose seaweed, Thermus aquaticus, tDNA, plasmid DNA, vector, SAL1 is a restriction and a nucleus. Okay, so SAL1. So you might have heard the term SAL1. Okay, so SAL1 is a restriction enzyme which is present on PBR322 at a tetracycline. Yeah, so agarose, seaweed. Of course, agarose can be extracted by using seaweeds. So seaweeds in the Gracilaria ಮತ್ತು ಜಿಲೇಡಿಯಿಂದ isolate ಮಾಡ್ತೇವೆ. So that is right. Then a Thermus aquaticus, tDNA. Okay, so tumor inducing DNA. So Thermus aquaticus do not have this tDNA. So tDNA, tumor inducing DNA, is present only in Agrobacterium tumefaciens. So this is the right answer. Plasmid DNA can be used as a vector. That's right. Then SAL1 is a restriction endonucleus present in PBR322. Okay, so illa sari ide tappagirode yadu andre B. So based on this, B is the right option for this question. Okay. Yeah. Next, move on to the next question. Yeah. So it's regarding PBR322. So PVU2 something is messed up. So extra banbade PVU2. Yeah. So PVU2 for ROP. So it should be here. Yeah. So the given figure shows the E. coli cloning vector showing the restriction site. Some parts of the labeled A to D. Choose the option showing the correct labels. Okay. So ಇಲ್ಲಿ PST1 and PVU1 are present on one. That is what is C. So remember, ಇಲ್ಲಿ tetracycline ಇದೆ ಅಂತಂದ್ರೆ ಈ ಕಡೆ ampicillin ಇರುತ್ತೆ. So C must be ampicillin. So okay, C must be ampicillin. Two options were there. So you can eliminate these two. Okay. Then A must be echo R1. So echo R1. So you need to remember the structure of PBR322. So A must be echo R1. So this is the right answer. Then B. So BIMH. Okay. Bacillus amarula liquefaciens. So here it is the SAL1 illirutte. So PVU2 on ROP, replication of plasmid can be controlled by this ROP region. So ROP region mele ಒಂದು restriction enzyme irutte, that is PVU2. Okay, so remember PVU1 and PST1 are present on tetracycline resistant gene. BMH1 and SAL1 they are present on PVU1 and PST1 present on ampicillin, BMH1 and SAL1 present on tetracycline resistant gene. If you use SAL1, ಕೇಳಿಸ್ಕೊಳ್ಳಿ, ಇದೊಂಥರ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ನಲ್ಲಿ we can frame the question. If you use the SAL1 in PBR322, transformants can be selected by growing on. ಅವಾಗ ನಿಮಗೆ ಗೊತ್ತಿರಬೇಕು SAL1 is present on tetracycline. If you use the SAL1 means it cut at a tetracycline, so it loses the resistance against tetracycline. The transformants can be selected by growing on ampicillin. So you need to remember that one also. So remember, okay, so ಎರಡು restriction site, so antibiotic resistant gene ಅಲ್ಲಿ ಎರಡು ಕಡೆ ಎರಡು restriction enzymes ಇದೆ. You need to remember both PVU1 and PST1. Okay, so ಇದು then BIMH1 and the SAL1. Okay, so ಎರಡು ನೆನಪಿಟ್ಕೊಂಡಿದ್ರೆ ಸಪೋಸ್ a geneticist ಅಥವಾ during recombinant DNA technology a scientist used BAMH1 enzyme to clone the DNA. Okay, so transformants can be selected by growing these bacteria on ampicillin. ಯಾಕೆ ಇಲ್ಲಿ ಕಟ್ ಮಾಡಿದ್ರೆ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಹೋಗ್ಬಿಡುತ್ತೆ, so they need to be isolated by using ampicillin. If a scientist use PVU1, transformants can be selected by growing on tetracycline. So like that you need to remember. So you need to remember these two, PVU1 and PST1, BMH1 and SAL1. So totally how many restriction sites are present on PBR322 andre count ಮಾಡಿ 1 2 3 4 5 6 7 and 8. So totally 8 restriction sites were there. Okay, so 8 different restriction enzymes can be used to clone the DNA by using this PBR322. So remember the structure and the sites of restriction enzymes very carefully. So of course D ಹೇಳಿಲ್ಲ, so D is the ORI site, so replication site, origin of replication site. Okay, so move to the next question. Yeah, so which one of the following option is correct for ABC marked with the given diagram of a recombinant DNA technology? So recombinant DNA technology with the diagram ಇದೆ. So cutting both the foreign DNA and vectors, DNA splicing point ಅಲ್ಲಿ foreign DNA ಇದೆ, ಜೊತೆಗೆ ಇಲ್ಲಿ ಪ್ಲಾಸ್ಮಿಡ್ ಇದೆ. ಸೊ ಏನು ಆಗಿರಬಹುದು? ಓಕೆ, ಸೊ ಯಾವುದನ್ನ ಇದು ಮಾಡಿದ್ರೆ ಡಿ ಎನ್ ಎ ಫ್ರಾಗ್ಮೆಂಟ್ ಸಪ್ರೇಟ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೊ ಯು ನೀಡ್ ಟು ಕಟ್ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಆರ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಆಪ್ಷನ್ ಇದೆ, ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. ಹೇಗೆ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ, ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. ಸೊ ಇಲ್ಲಿ ಬರೀ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್, ಇಲ್ಲಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್. ಸೊ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ದ ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಸೊ ಬರೀ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಅಂತಿದ್ರೆ ಬರೀ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲ. ಸೊ ಪ್ರಿಫರಬಲಿ ಇಟ್ ಶುಡ್ ಬಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್. ಆಗ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ಇಲ್ಲೂ ಕೂಡ sticky ends ಬಂದಿರುತ್ತೆ, ಇಲ್ಲೂ ಕೂಡ ಸೇಮ್ ಕೈಂಡ್ ಆಫ್ sticky ends are going to be produced. Okay, so after that we can join both of them by using one enzyme. ಎರಡನ್ನು ಜಾಯಿನ್ ಮಾಡ್ತಿರೋ ಎಂಜೈಮ್ ಯಾವುದು ಅಂದರೆ ಲೈಗೇಸ್ ಎಂಜೈಮ್. ಸೊ ಬಿ ಶುಡ್ ಬಿ ಲೈಗೇಸ್. ದೆನ್ that now this is called rDNA. So ಒಂದ್ಸರಿ foreign DNA introduce ಆಯಿತು ಅಂದರೆ so it's become rDNA. So this rDNA can be introduced into the bacterial cell by making the bacterial cell competent. So how to make the competent we are already discussed. By heat shock method we can make the bacterial cell competent. Now when the DNAs get taken up by this bacteria now become transformant. Okay, so this transformant can be selected and cultured in a bioreactor to get the product. So it is transformation. So these are the options. Okay, so D is the right option. So here is A, restriction enzyme, endonuclease enzyme. B is the ligase enzyme. C is transformation. Okay, so 3 options. Okay, next question. So it's regarding a bioreactor. The given figure shows a simple stereo tank bioreactor with their parts labeled as A, B and C respectively. What are those A, B and C? Okay, so bioreactor ಅಲ್ಲಿ ಇಲ್ಲೊಂದು ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ. So it is like this. ಇಲ್ಲಿ ಬ್ಲೇಡ್ಸ್ಗಳಿದೆ. ಸೊ ಇದು ರನ್ ಆಗಲಿಕ್ಕೆ ಇಟ್ ಶುಡ್ ಬಿ ಎ ಮೋಟಾರ್. ಸೊ ಎ ಶುಡ್ ಬಿ ಮೋಟಾರ್. ಓಕೆ, ಸೊ ಎರಡು ಕಡೆ ಇದೆ ಎ ಮೋಟಾರ್, ದೆನ್ ಎಲಿಮಿನೇಟ್ ದಿಸ್. ಓಕೆ, ದೆನ್ ಸಿ ಬರೋಣ. ಸೊ ಸಿನಲ್ಲಿ ಎರಡು ಆಪ್ಷನ್ಸ್ ಇದೆ, ಪಿ ಎಚ್ ಕಂಟ್ರೋಲ್ ಮತ್ತು ಸ್ಟರೈಲ್ ಏರ್. ಓಕೆ, ಸೊ ರೈಟ್, ಸೊ ಸ್ಟರೈಲ್ ಏರ್ ಡಿ. ಇಲ್ಲಿ ಸಿ ಏನಿದೆ, ಸೊ ಸಿ ಈಸ್ ಅ ಫೋಮ್ ಬ್ರೇಕರ್ ಆ್ಯಕ್ಚುಲಿ. ಸೊ ಸಿ ಈಸ್ ಅ ಫೋಮ್ ಬ್ರೇಕರ್. ಸೊ ಪಿ ಎಚ್ ಕಂಟ್ರೋಲರ್ ಇಲ್ಲಿದೆ. ಸೊ ಬಿ ಈಸ್ ಅ ಪಿ ಎಚ್ ಕಂಟ್ರೋಲ್, ಸಿ ಈಸ್ ಅ ಫೋಮ್ ಬ್ರೇಕರ್. ಅದನ್ನು ನೊರೆ ಬರ್ತಾ ಇರುತ್ತೆ, ನೊರೆನೆಲ್ಲ ರಿಮೂವ್ ಮಾಡಲಿಕ್ಕೆ ಫೋಮ್ ಬ್ರೇಕರ್ ಇಸ್ ದೇ. ಸೊ ಇಟ್ ಈಸ್ ನಾಟ್ ಎ ಪಿ ಎಚ್ ಕಂಟ್ರೋಲರ್. ದೆನ್ ಡಿ ಈಸ್ to pass the sterile air. Okay, so that's it. So right option is A. So A is the right option. So go through each and every diagrams in the NCERT book and their labels. So based on this also, they may ask the questions. Okay. Yeah. So the figure given below shows 3 steps A, B, C. Okay, so diagram is missing. So let me write that diagram. Okay, so this is B. So here is the diagram. So with the enzyme. Okay, so let me label the diagram ABC. So here is A, B, C. Okay, so the figure given here shows the 3 steps ABC of polymerase chain reaction. Select the options giving correct identification together with what it represents. So what does it A and B, C represent? So there are different options. So you need to choose the right one there. So which one is right antele. So B, denaturation at a temperature of about 98 degrees centigrade, separating the 2 DNA strands. So B ಯಾವ್ದು ಕೊಟ್ಟಿದ್ದಾರೆ. So actually A is denaturation, but temperature ಕೂಡ wrong ಇದೆ. So it is not the option. Then A is a denaturation at a temperature of about 50 degrees centigrade. Okay, of course here is a denaturation, but it is not 50 degrees centigrade. So it is not that option. See, extension in the represent of a heat stable DNA polymerase. So polymerase enzyme extending DNA replication ಮಾಡ್ತಾ ಇದೆ. So it is the right. Then A annealing with the 2 sets of primers. So A ನಲ್ಲಿ annealing ಆಗ್ತಾ ಇಲ್ಲ. So the right option is C. Okay, so their diagram is missing. So this is the diagram. Okay, yeah, that's it. So hope you got it with respect to PCR steps. Okay. So next move to the next question. Yeah. So DNA fragments are separated using gel electrophoresis. Because DNA is pulled through the gel towards the negative end of the field. DNA is pulled through. No, it is not pulling. Okay. So because the larger DNA fragments move faster. No. To identify and isolate DNA fragments. Yeah, that is the step. Isolation of DNA fragments. To synthesize DNA for cloning. That is PCR. So right option is C. Okay. So to identify and isolate the DNA fragments, you go for gel electrophoresis. Okay. Yeah. Next. Imagine a gel through which DNA fragments have moved response to an applied electrical current. The band on this farthest from the band on this is farthest from the top, that is from the place where the DNA fragments are added to the well, represent shortest fragment of DNA, longest fragment of DNA, restriction enzyme used to cut the DNA into fragments, ligase used to bind the DNA fragments together. Okay, imagine a gel electrode through which DNA fragments have mood response or applied electric current. Okay, the band on this is the farthest from the top, that is from the place where DNA fragments were added. Okay, so ಈ ಕಡೆಯಿಂದ add ಮಾಡಿದ್ರೆ DNA fragments were added to the well, represents. Okay, so ಇಲ್ಲಿ ನಾವು DNA add ಮಾಡ್ತಾ ಇದ್ದೀವಿ ಅಂದರೆ shortest fragment of DNA, longest fragment of DNA. ಸೊ ಇದರ ಅರ್ಥ ಅಲ್ಲಿಂದ ದೂರದಲ್ಲಿ the band on this farthest, ಇಲ್ಲಿಂದ farthest ಅಂದರೆ ದೂರದಲ್ಲಿರೋ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಯಾವ ಥರ ಇರುತ್ತೆ. ಅಫ್ಕೋರ್ಸ್ if you look into the property of separation, principle of separation, ಸೊ ಎಲ್ಲಿ ಡಿ ಎನ್ ಎ ಲೋಡ್ ಮಾಡಿರ್ತೀವೋ ಅಲ್ಲಿಂದ ಫಾರ್ದೆಸ್ಟ್ ತುಂಬ ದೂರದಲ್ಲಿರೋ ಫ್ರಾಗ್ಮೆಂಟ್ ಯಾವುದು, ಇಟ್ ಶುಡ್ ಬಿ ಶಾರ್ಟೆಡ್ ಫ್ರಾಗ್ಮೆಂಟ್ ಆಫ್ ಡಿ ಎನ್ ಎ. ಓಕೆ, ಸೊ ಕ್ವಶನ್ ಸ್ವಲ್ಪ 2-3 ಸರಿ ಓದ್ಕೊಂಡು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ದೆನ್ ಯು ಆರ್ ಏಬಲ್ ಟು ಗೆಟ್ ವಾಟ್ ದೇ ಆರ್ ಆಸ್ಕಿಂಗ್. ಓಕೆ ಓಕೆ ಯಾ ನೆಕ್ಸ್ಟ್, biolistic is suitable for DNA fingerprinting, dissolving pathogen, transformation of plant cells, constructing recombinant DNA molecule. Yeah. So it is to transformation of plant cells. So for that purpose we can use them. Okay, so that's it. So this is about the biotechnology principle and process chapter. Okay, yeah. So thank you.